Hello, ladies and gentlemen, and some students. Thank you for joining the Science Symposium. For the afternoon session is about life science section. We'll be focusing on transformative science for human biology and health. Now, I would like to introduce with you Professor Tony Shu-Kung-Mok as the section's convener. He is Chairman, Department of Clinical Oncology, Li Shufan Medical Foundation Professor of Clinical Oncology, Chinese University of Hong Kong, Honorary Secretary of the Board of Directors and member, Hong Kong Academy of Sciences, member, Program Committee of the Future Science Prize Week 2024. Please give big hands to Professor Mok.
Thank you. Great pleasure of mine to welcome all of you here this afternoon in the life science session. I'm most excited to see not just the professor, but the young student to be here because you are our future. So as you probably know, life science is one of the most important disciplines of applied sciences in the 21st century. Not that we only study the life phenomena and also review the law of life and activity and the essence of life. Indeed, we're surrounded by biosciences right at this moment. However, we're all facing major challenges in the society this day. Different disease, cancer, cardiovascular disease, food shortage, energy crisis, and environmental pollution. And to solve all these problems, we need bioscience and biotechnology. So today, with great pleasure, we have 4 major speakers, keynote speakers from different disciplines and different parts of the world on very interesting topics. And I here announce the beginning of the life science section, the transformative science for the human biology and health. With that, I would like to introduce our first speaker, Professor Anne Ferguson Smith, CBE, who is also an Arthur Bethel Professor of Genetics, the ex-Vice Chancellor for the Research and International Partnership at the University of Cambridge, and also the Executive Chair of the Biotechnology and Biological Science and Research Council. More importantly, she's also a fellow of the Royal Society and also the Academy of Medical Science. And she's going to talk to us about interesting topic, epigenetic, what is it, and why is it so important. Anne, may I? Thank you.
Hello, everybody. It's a real pleasure to be here at this fantastic symposium. And this morning was wonderful getting to hear about science outside my own discipline. It's a real luxury to sit and learn about catalysis and longer batteries, and it really exemplifies the value of hanging out with different disciplines and thinking about how what others do can influence how you think, and how what you do can influence what others think. And so it's a real pleasure to be part of this symposium, and thank you for having me here. So I'm going to talk about epigenetics. And the problem really begins really within the nucleus of a cell. So the nucleus of a cell, of a human cell, is about 6 microns in diameter, and one has to squeeze 2 to 3 meters of DNA into that nucleus. And that DNA is not really, therefore, how we think about it as a double-strand helical structure. Of course, it is that, but it has to be packaged and organized in such a way that it fits. And so what you see here is the DNA, which is modified with DNA methylation here. I'll come back to that in a minute. And it's wound around a structure called a nucleosome, which is made of 4 core histones, in fact, 2 copies of each of the 4 core histones, and these histones have tails. These tails can carry chemical modifications, and that influences how this chromatin is wound into the beautiful structure that is the chromosome. And this chromosome, in humans, there are 26 pairs of chromosomes, and they're packaged and bundled into this nucleus. But it's not just about packaging. This process is highly regulated. And it's regulated in much part by epigenetic modifications, these chemical modifications that sit on the DNA and the histone proteins. So epigenetics, what is epigenetics? There's lots of people who spend a lot of time trying to define it. I don't, but because there are others who do it much better than I. And so over the years, it has had a number of definitions. My favorite one is probably the one most recently from Adrian Bird, who defines epigenetics as the structural adaptation of chromosomal regions so as to register, signal, or perpetuate altered activity states. But for me, really, epigenetics is about the modifications, chemical modifications to DNA and chromatin and their influence on genome function. And the modifications that we think about the most are DNA methylation, this mark that sits on the cytosine when it's next to a CG in mammalian genomes. And then, obviously, it's symmetrical, so it would sit on a C opposite a G here. And the other modifications are these post-translational modifications to the core histones that influence how chromosomes are packaged. And they contribute to, epigenetic modifications evolve to do three things. Can I ask, I can't see the bottom of that slide. Can you see it, the third bullet point underneath? Good, okay, because I can't see it from here. So epigenetic modifications evolve to really do three things. The first is to contribute to the functional architecture of chromosomes. So what does that mean? These chromosomes have constrictions in the middle called centromeres that are really, really important for the pulling apart of chromosomes into daughter cells during cell replication. And epigenetic modifications act in the regions of these centromeres to make sure that there's strong, robust structures that enable proper segregation of chromosomes. The architecture of the chromosomes is important for that process. The second thing is that they contribute to the dynamic regulation and heritable maintenance of gene expression, particularly during development, and I'll talk a little bit about that. And thirdly, and very importantly, something that we don't think about very often, but they actually have evolved to manage the half of our genome that is repetitive, and I'll talk about that also. And I know that Hai-Fan Lin will also talk about the management of repetitive elements by epigenetic states too. So, the epigenome, so while we have one genome in all of our cells, each of our cells has pretty much the same DNA content, we have lots of epigenomes. And that's because the epigenome is both dynamic and stable, and importantly is cell type specific. So for example, in neurons, in brain cells, we need to organize our DNA in such a way that the genes that are required for neuronal function are expressed, are accessible to the machinery of the cells that turns genes on and off, and genes that are important for liver function, for example, are not. And so that's illustrated here, where you can, where you can see areas, for example, where a heart gene might be sitting here in a neuron. It's closed, it's modified, it's marked with repressive modifications that ensure that that chromatin is tightly packaged, whereas genes that are required to be expressed are in a more open state. And that is, of course, regulated by transcription. Gene expression is regulated by transcription factors, but those transcription factors are interacting with the epigenetic machinery, which is maintaining this gene in an open state. Similarly, in a cardiomyocyte, a heart cell, we have a heart gene that is marked with modifications that keep it open and adjacent to, for example, brain genes that have to be closed so that the wrong genes don't get expressed in particular cells. Epigenetic marks are generated and acted upon by enzymes and proteins that write the mark, that read the mark, and then can remove the mark in contexts where the epigenetic state is dynamic. And many of these readers and writers and erasers have been well characterized, and some of them have been, the expression of some of them have been perturbed in cancer cells, for example, resulting in abnormal epigenetic modifications, epigenetic states in cancer. And furthermore, some of these readers have been targeted as therapeutic agents for drugs in particular cancers. So there is a growing field in epigenetic therapies, particularly in cancer. But importantly, And I'm going to focus predominantly today on DNA methylation because that's the mark that we work on the most. It is a mark that is stably maintained during cell division. And that's because a methylation at a CG, the enzymatic machinery that places new methylation marks on before there was none, recognizes that newly synthesized strand of DNA with the CpG dinucleotide, recognizes it across from the old strand that is already methylated, so the enzyme recognizes so-called hemimethylated DNA and puts methylation on. So it sees the methylation on the old strand and puts it on the new strand. And that means that there is a mitotic heritability. There is the ability to inherit a methylation state from a parent cell to a daughter cell. Now that is different from heritability from one parent generation to children, to offspring, which is going through a germline. And that is a different process and a different mechanism. And it's very hard to imagine epigenetic inheritance from parent to offspring because in mammals, the epigenetic marks are erased and reconstructed from one generation to the next. So the epigenetic memory that is established in one parent is generally lost from one generation to the next. And that's illustrated here on the next slide, where you can see, which is a graphical representation of the dynamics of DNA methylation in the two periods where epigenetic marks are erased and reconstructed. The first is during the development of eggs and sperms. These are primordial germ cells, cells that are going to become the eggs and the sperms. And as they develop, this is data from the mouse, by about little after mid-gestation in a mouse embryo, all the epigenetic marks, including DNA methylation, have been erased. The germ cell, the nucleus, the DNA is epigenetically naked. And then in the male germline and in the female germline, in the making of sperm and in the making of eggs, new marks are placed on in order that the genes that are important for making sperm and the genes important for an oocyte function are properly regulated. And then upon fertilization, lo and behold, the whole thing happens again, and the male germline loses its epigenetic marks actually quite quickly, and the female germline loses her epigenetic marks. The female chromosomes lose their epigenetic marks a little bit more slowly. And then, around the time the embryo implants in the mother's uterus, they start to get those marks back again, and those marks occur in a tissue-specific and cell-type-specific manner in the way in which I explained to you in the very first slide. So if we have this erasure and reconstruction not once but twice in mammalian development, it begs the question about epigenetic inheritance, and that's what I'm going to talk about for the last part of the last half of my talk. So really, the idea here is that the question really is, to what extent are methylation states that are established in one generation transmitted to the next? Can they be? If I've told you that they're generally erased and reconstructed, a lot of interest in epigenetic inheritance. How can that happen if these marks are erased and reconstructed? And then the next question, which is a pervasive question in the field, is that does the environment talk to the genome via the epigenome? And if it does, can that change, can that influence be perpetuated across generations? Remember, the template for our inheritance is our DNA, is our genetic material, but is there a role for epigenetics? And so to answer, I'm not going to answer those questions completely, but I'm going to be provocative and explore them with you for the rest of my talk. So I'm going to talk about my two favorite models of so-called epigenetic inheritance. One is a process called genomic imprinting, which I've worked on for many years, which is a true model of epigenetic inheritance, and we understand how that works. And then the second model I'm going to talk to you about is associated with these repetitive sequences, these regions of the genome that actually look like viruses and have the potential to mobilize and hence are targeted by the epigenetic machinery. Let's explore a little bit about their epigenetic inheritance. And this is to remind, this sentence at the bottom about the value of inbred laboratory mice is to remind me to tell you that I'm a fundamental, basic researcher. I'm not working on translation. My interests are in making conceptual advances in our understanding of molecular genetics. And that particularly for epigenetic studies, the laboratory mouse is a really good model for looking at this, because you can breed laboratory mice such that they are genetically homogeneous. If you were to do this kind of things in people, all of you in the room have different genotypes from each other, and many of the epigenetic changes that one would what one finds in you will be because they're sitting on top of perhaps a sequence polymorphism where the CPG that should be that is methylated in some of you has been changed to a TG and therefore isn't methylated, and that's a genetic rather than an epigenetic change. It's a genetic change that underpins an epigenetic change. And by using inbred strains of mice that are genetically homogeneous, we get away from that genetic variation and makes this a very good model for studying both epigenetic variation and epigenetic inheritance. So I'm going to start to talk with genomic imprinting. And to do that, I want to go back to basics, because I know that there are physicists in the room, and to remind you that upon fertilization, the embryo inherits one set of chromosomes from the egg and one set of chromosomes from the sperm. And I've illustrated these here as, as a blue set from dad and a red set from mum. So 2 copies of every chromosome, apart from the sex chromosomes. That's not relevant at the moment if you're a male. I'm talking here about non-sex chromosomes. So what is genomic imprinting? Well, when I was a graduate student, I read the most amazing piece of work. And it was published in 1984, which is, and so this year, this discovery is celebrating its 40th anniversary. And it was this experiment here done independently by 2 researchers, 2 embryologists, one called Azim Sarani in Cambridge, and the other called Davor Salter, which work that he did when he was in Germany. And what they discovered, oh no, maybe he was in the US when he did the original experiment. So what they did was they took a newly fertilized mouse egg and they removed the male pronucleus. This is the bag of chromosomes that came in from the sperm. They whipped them out and replaced them with a second set of chromosomes that came from mum. Alternatively, they whipped out the bag of chromosomes that had just come in, that are just coming in from the egg, and replaced them with a second set of chromosomes that had come from dad. So they generated embryos that had the normal chromosome complements in terms of numbers. So they were a normal diploid set of cells, but they had altered the parental origin of the chromosomes. So so what they discovered, and here's the nice control in the middle, which has the normal set of chromosomes from dad, set of chromosomes from mum. And what they discovered was that these 2 experimental embryo types could not make it to term. They failed, despite having the normal genetic chromosomal constitution. What happened here is if you have 2 copies from dad and none from mum, you make a lot of extra embryonic tissue, and very rarely do you get an embryo at all. This is the embryo, and it's very retarded and very underdeveloped. And it's, but as I say, often it's just not there at all. And on the diploid maternal or gynogenetic conceptus, you get a rather nice-looking embryo proper. You get very pathetic extra embryonic tissues, but the embryo itself is very small. And these embryos, as I say, will die. And so what this said, what this told us, was that actually being diploid is not enough. You need both a maternal and a paternal set of chromosomes in order to develop as a mammalian embryo. It also told us that the maternally inherited chromosomes function differently to the paternally inherited ones. Now, this is what blew my mind when I was a graduate student, because Mendelian genetics told us that, you've got these genes from mum, these genes from dad, and you have 2 copies, and they're both either expressed or repressed at the same time. This was telling us that these chromosomes were functioning differently. It also suggested that there was something about the maternally inherited chromosomes that looked different from the paternally inherited ones, even though they were genetically pretty much the same. And then finally, it suggested that there was a memory, some kind of memory of parental origin that was conferring these functional differences on these two chromosome homologs in the newly fertilized, in the embryo during embryogenesis. This is not a mouse thing. This happens in humans as well. An androgenetic conceptus in humans is a complete hydatidiform mole, which is a diploid bipaternal contribution. It resembles a malignant mass, and like the mouse, it has an excessive cytotrophoblast proliferation. The extraembryonic cells are overproliferative. And the diploid maternal is known as an ovarian teratoma, which is a germ cell tumor of the egg. So it's a sort of activation of the female chromosomes. It's usually benign, and it contains tissue lineages such as bone and hair and teeth. So it has embryonic components to it rather than extra embryonic components to maternal genomes. So we now know that this process of so-called genomic imprinting is a process that causes genes to be expressed differently depending on whether they've come from the mother or from the father. So these two chromosomes, they look the same genetically. They both contain gene A, and although the sequence of gene A is to all intents and purposes the same, apart from polymorphisms between males and females, only this one that's been inherited from mum is expressed, the other one is repressed. Likewise, another gene nearby even is expressed from the paternally inherited chromosome and not from the maternally inherited one. Whereas most other genes are either expressed from both or repressed from both. They behave in the same way. So over the course of many years, we and others have tried to understand this mechanism, to try and understand what it is that's different about these two parental chromosomes. We know that this is due to differential epigenetic modifications. So the DNA may be the same, but the epigenetic marks are different on the two parental chromosomes. So what do these genes do, these special imprinted genes? Well, they're very important for development. They're very important in prenatal growth and placentation. They play a particular role in the development of particular lineages, such as bone and cartilage. This is a developing placenta. We know that they play a role in postnatal and prenatal, in fact, brain and behavior. Imprinted genes are important in the regulation of adult neurogenesis, for example. And we know that they're important for postnatal life course events. So this is a model whereby the mice are not, the mice don't do non-shivering thermogenesis properly. They cannot control their temperature properly, and that's because of the altered dosage of imprinted genes. So they play a role in fat metabolism and other postnatal physiological processes. In humans, you can have alterations in these imprinted genes, and they result in imprinted syndromes. Imprinted genes generally hang out in clusters. There's multiple imprinted genes alongside each other, and these clusters get perturbed, or you get when they get perturbed, or you get loss of imprinting, you end up with imprinted syndromes, and there are 8 human imprinted disorders that have been characterized to date, and they are generally associated with 3 types of mechanisms. Either an alteration in the dosage of the parental chromosomes, so you can see here that if you have 2 paternal chromosomes instead of one from mum and one from dad, you get a double dose of gene A, and that's bad for you. You can also have a deletion, and if the deletion happens in the active copy of gene A, then you're basically not making any of gene A, or you're functionally haploid through imprinting. Alternatively, if you were to lose this copy, nothing would happen because it's the paternal one that's being expressed. An example of an imprinted gene actually is the insulin-like growth factor 2 gene, which is very, very important for development when you're an embryo. And it's only expressed from the copy that you inherited from your dad. The copy that you got from your mum is silent. And so if something like this happened, and you ended up with 2 copies of your dad's chromosome and none from your mum, you'd be making a double dose of this insulin-like growth factor 2 gene. You'd be really big, and you'd also have an increased incidence of childhood cancers. So that's the sort of relevance of imprinting in human contexts. So how does the transcriptional machinery, how does the gene expression machinery tell the difference between these 2 parental chromosomes and express one and not another? Well, it's because the genomic imprinting is actually a DNA methylation mark that is placed differently on the imprinted chromosomes in the egg compared to in the sperm. So, and there's about 20 imprints that are established in the egg. This is DNA, a filled lollipop means methylated DNA in the vicinity of this gene. And likewise, in the sperm, there are actually only 3 imprinting control regions in the sperm. They're never at the promoters of genes. They're usually in the middle of the, in between genes. And upon fertilization, you end up inheriting an unmethylated copy from dad and a methylated copy from mum. And DNA methylation is generally associated with repression. And so this copy is active and this copy is repressed. And likewise, you inherit a methylated copy from the dad, an unmethylated copy from the mum. You're differentially methylated between the 2 parental chromosomes. So imprints are regulated by germline-derived differentially methylated regions, which are heritably maintained in offspring after fertilization. And often a single differentially methylated region, an imprinting is an imprinting control region for lots of genes in the cluster. So how can that happen when I told you that, that these epigenetic marks are erased and reconstructed? How can you remember, how can you have a memory, an epigenetic memory of these parental imprints in a context where upon fertilization everything is lost? Well, what we know is that it's not the germline methylation that is the essential component of epigenetic inheritance at imprints, because actually there's lots of regions of the genome that are differentially methylated between eggs and sperms, but it's the retention of that methylation after fertilization that is absolutely the key. And that is regulated by, so that is illustrated here, where you see a context where all the rest of the genome is losing its marks, but imprinted germline DMRs are retaining theirs. And they're retaining them through the presence of 2 proteins called crab-zinc finger proteins that bind to these methylated regions and target a continuous acquisition of methylation, so the memory is retained. And we know that because when we mutate these 2 proteins in the mouse, the imprinting memory is lost. The animals lose their memory of parental origin. I want to say a little bit about these proteins because people don't really know very much about them, and they're really interesting. They're a family of DNA binding proteins that are unique to vertebrates, and there's hundreds of them. They recruit the repressive chromatin complexes, resulting in the writing of repressive histone modifications and DNA methylation. And they're very different between different species. Of the 300 or so, only 80 of them are common to mouse and human, and that's actually quite unusual for important regulators. For imprints, these are they're 2 methylation-sensitive crab-zinc finger proteins. They bind to methylated DNA, but most of them don't. Most of them are actually targeting methylation to regions that are previously unmethylated. And what they really evolved to do was to target the 50% of our genomes that are highly repetitive, look like viral sequences, and have the ability to move around. And so that brings me to the second part of my talk. For the last 5 minutes or so, I'll tell you about variable DNA methylation at transposons. I'm going to have to whiz through this. So this is to just tell you that retrotransposons are substantial. So this is a whole bunch of different organisms. They all have some kind of repetitive sequences within them, but the mouse and human have, close to, 40 to 50% of their genomes are made up of these repetitive sequences. And they're targeted after fertilization by the repressive chromatin machinery and DNA methylation via these crab-zinc finger proteins. And I think you'll hear from Haifan how these particular sequences are treated in the male and female germlines, well, in the germlines in particular. I don't know if he's going to talk about that or not, but he might. But many of these elements have lost the ability to mobilize, so they're actually just sitting there, they're broken, but yet they're still targeted by the epigenetic machinery. So the second model that I'm excited about in this context is a classic mouse model called agouti viable yellow. So these 6 mice look very, very different, but they're actually genetically identical. So their coat colors are ranging from yellow to brown and everything in between, yet they're genetically identical. So what's happened here? Well, one of these repetitive sequences, called an IAP, has inserted upstream of the coat color gene and provided a regulatory sequence that inappropriately turns this gene on. And when you turn this coat color gene on too much, you get a yellow mouse. And when you don't turn it on at all, you get a brown mouse. And the ability to turn the gene on depends on whether this retrotransposon has been methylated or not. So these yellow mice have no methylation on their transposon, yet the brown ones, called pseudoagouti, have very high methylation. And the mottled mice have levels of methylation in between. So here we have a visible phenotype that is regulated not by genetics, but by epigenetics. And this mouse model is a very exciting one because it's quite sensitive to environmental factors. So you can feed mice methyl donors, and you increase the methylation at these, and you shift the coat color. You can also treat pregnant females with the endocrine disruptor bisphenol A, and you shift the coat color towards yellow. So it's a model of environmental compromise. In addition, and most importantly, it displays transgenerational epigenetic inheritance, apparently. And what you see here is that yellow mice are much more likely to have yellow offspring, and brown mice are much more likely to have brown offspring. And I told you that the color is conferred by the epigenetics, not the genetics. Therefore, that propensity is epigenetically inherited. So how does that happen when I told you that the epigenome is erased and reconstructed? What happens at agouti viable yellow? Well, we know that the methylation is actually treated the same way as the rest of the genome, not like imprints. So in this case, the epigenetic memory is erased and reconstructed. And we actually, in contrast to imprints, we don't know the mechanism of epigenetic inheritance at this mutant. We wanted to ask whether this particular agouti viable yellow was special, or whether any of the other 10,000 transposons of this particular class behaved in this way too in the genome. So we did a genome-wide screen for repetitive elements that behaved like the one at agouti viable yellow, and we found about 100 of them. Some of them are influencing genes, some of them are not. They're very young. They've recently jumped into the mouse genome. We know that the mechanism is conferred by these crab zinc finger proteins who don't do it very well, and as a result, you get inter-individual variation in methylation. And we wanted to ask whether it was these elements were environmentally sensitive and what their patterns of inheritance was. And I'm conscious of time, so I'll be very quick to say, basically, to cut a long story short, they're not environmentally sensitive. They don't respond to the environment. So agouti viable yellow seems to be special in that context. And that's just the methyl donor diet, so showing no effect. And that heritability is the exception rather than the rule. So in fact, most of them don't show any epigenetic inheritance. So what I've told you really, I hope, is that epigenetic modifications are important. They sit upon our genetic material and they influence how genes are expressed or not. But importantly, in mammals, they are erased and reconstructed between generations, which means that it's difficult to inherit an epigenome from one generation to the next, except at very special places where it's very highly regulated, such as genomic imprints. Epigenetic modifications evolved to silence these repetitive sequences in our genome, to target these sequences, and they do it usually very well, but some are not fully silenced and can influence adjacent gene expression, like I showed you for agouti viable yellow. So these are epigenetic effects. Agouti viable yellow is an epigenetic effect that causes differences between individuals. Not genetic effects, but epigenetic effects. And that, I think, has non-genetic implications for disease, and actually even for normal outcomes that might explain why some of us are different from each other, not for genetic reasons, but because our repetitive sequences have variable epigenetic states that influence the expression of adjacent genes that cause differences between us. And we have evidence now in mouse that I didn't have a chance to talk to you to say that, in fact, this is true, and it's quite important, for example, for metabolic phenotypes and other phenotypes that are relevant for complex disease. So I'll stop there and thank the people in my team who've done the work. The work that I talked about is predominantly being done by this team here, predominantly Kevin Costello and Nozomi Takahashi. And I thank our funders and our collaborators who worked on the environmental modulation stuff with us. And I thank you for your attention.
Thank you, Anne, for the marvelous talk on epigenetic. Now I know who I am, why I am is actually related to my epigenetic. Hopefully one day they can modify my epigenome a bit better to make me a bit smarter. So with further ado, I would like to introduce the next speaker, who is Professor Euan Ashley, the Associate Dean of the School of Medicine, also the Roger and Joelle Burnell Professor of Genomic and Precision Health. He's also the Professor of Medicine in Cardiovascular Medicine, of Genetic, and also of the Biomedical Data Science, and also by courtesy of Pathology. So he's a professor of many things. More importantly, I read his CV, and one of his lifelong interests is looking at the health impact of the Scotch single malt whiskey, which I would very much like to participate in his experiment. So he is going to talk to us about realizing the potential of biomedical AI. Euan, thank you.
I am very grateful to Anne for tuning your ears to a Scottish accent just in time for the second Scottish speaker of the day. Thank you for the invitation from the committee to join you today to talk about artificial intelligence. We are in the middle of a revolution in machine intelligence that will touch very many aspects of our lives, and I'm going to focus today, of course, on how that will affect us in science and how that will affect us in medicine. Here are my financial disclosures. Many of the images, including the one you saw on the last slide, are generated by AI. I think, surprisingly, this image is not generated by AI and comes from the James Webb Telescope. This is the Carina Nebula, 7,000 light years away. But I think the quote from Arthur C. Clarke is pretty relevant to the way many of us are thinking about artificial intelligence today. Some of the things it can do are indistinguishable from magic. And AI has surpassed humans at a very large number of tasks. And more importantly, the rate at which humans are being surpassed is increasing. If you look at this graph from the late 1990s to now, the slope of these lines as AI beat us at handwriting recognition, for doctors that's not surprising maybe, language understanding, reading comprehension, image recognition. Even since this graph was made in 2022, AI has surpassed not just grade school math, but high school math and now college math. So the rate at which it is improving is remarkable and is increasing. Since 2023, these multimodal AIs can understand and explain complex images and diagrams. It can break down an image like this and explain it to you in multiple steps. It can even make me multilingual. I don't know if the audio will. You can't hear the audio. Okay, you just have to believe me that this is me speaking in Japanese. That doesn't really work with the audio, unfortunately. Well, anyway, I have Mandarin in there as well, as Polish and Spanish. And the most freaky one, actually, of all, is me speaking with an English accent, which is quite strange. Anyway, I'm very sorry that you've missed that. I guess the audio later will not work either, but never mind. AI can also remarkably talk to animals, including whales, and even plants. This is a paper in Cell from just last year, looking at the sounds emitted by plants under stress, and it's essentially parsing them using artificial intelligence. Also, perhaps most remarkably, there are emerging properties of these artificial intelligence models where they appear to even have a theory of mind. So if you look at this little diagram shown here, just for a moment, I'll let you take it in with your human brain. Well, I think you see a cartoon where there are three adults and they think they agree, but they do not. They start talking and then they realize they don't agree and then they come to a compromise and at the end they all agree. The fact that we can understand that relates to the fact we are human and we understand what it is to interact with other humans. How does a large language model interpret this from the perspective of a human cognition? I say appears to have a theory of mind. These language models do not have a theory of mind, but they do a pretty good job of appearing to understand what it would be like to be inside a human brain. And I think it would have hardly escaped your notice that in the last couple of weeks, AI has swept the 2024 Nobel Prizes, both in physics, some debate about whether it really was physics, but certainly physics with Jeff Hinton, John Hopfield, and the chemistry Nobel for AlphaFold, Demis Hassabis, and John Jumper, and obviously David Baker there as well. Related to health and medicine, it's and somewhat more challenging for those of us who are practicing doctors. It turns out AI may be more empathetic than doctors. In this study of 195 randomly drawn patient questions from social media, both doctors and an AI responded, and then they were judged by both patients and doctors as to which were the more empathetic, and both the patients and the doctors judged the answers from the AI to be more empathetic than the answers from the doctors. So we may be out of a job yet. Although I'll come to some reasons why it may not be just today. Before we go forward, I want to go back. How did we get here? And the history of AI is actually probably quite a lot longer than most of you would remember, or perhaps even appreciate. The term artificial intelligence was first coined by John McCarthy and some other authors from MIT and Stanford in the 1950s. But even in 1950, you could trace back to when Alan Turing first published his classic paper outlining the Turing test in Cambridge. From then, through the 60s and early 70s, there were many breakthroughs, most of which were bi-coastal in the US between Stanford and MIT, actually shown in the back of this diagram. The reason you can see the Stanford campus through the middle of this history is because there was an AI winter for about 30 years. Very little happened, even arguably 40 years up to the early part of 2010, when suddenly we were in the deep learning era. And I'll pick up that history with the next couple of slides. You might remember the first time that an artificial intelligence algorithm was applied to the entirety of YouTube. It found two themes. One of them was human faces, and the other one was cats. Maybe not surprising to many of you who spend time on YouTube. But that entered us into the deep learning era, the era when you could ask a model what was contained within a photo, and it would use a series of deep learning algorithms to identify them, as shown here on the right. This was work pioneered by many, but including Jeff Hinton, one of those who just won the Nobel Prize and previously had won the Turing Award. And it really involved, for the first time, the bringing together of several different elements, including the training on very large data sets, the use of GPUs for that training, very deep models, through which backpropagation allowed optimization of those models in a way that may, or arguably may not, be similar to our human brain. This was around 2010, 2012. You may have seen this image before. Of course, it's a little bit of a test for a vision model and even for a human to tell the difference. Of course, in those days, it was a little different for those models to do that, a little difficult. These days, you just ask ChatGPT or similar, and it will actually lay out in a table for you which is a chihuahua and which is a muffin, which is helpful, obviously, in the real world. Maybe not. One of the most really important elements beyond deep learning, beyond the convolutional neural network that underlay those previous slides, was reinforcement learning, and this does not have audio, so you're missing nothing here. This is a classic paper from DeepMind, where they fed in basically Atari games, so some of the earliest games from the 1980s, I think, in computing, and basically fed it individual stills with different training episodes, and basically told it to maximize the score. And it basically came up then in the video domain, sort of in a temporal domain, with an approach to playing the game. As you'll see, it gets better after 400 episodes, training episodes, until at which point it learned something that humans eventually also learned about this game, and some of you in the audience may be old enough to remember this game, some of you may not. But after, I think it's 600 training episodes, you'll see, yeah, here we go. The agent actually finds and exploits a new strategy. I used to play this game, and it took a little while, but watch. It basically learns that if you fire the ball up in the back, it'll actually bounce up and down and give you many more scores. So reinforcement learning, you know, although it was here learning a computer game, and an older computer game at that, they actually were able to train the same model to play all the Atari games that had been released over the course of a decade and more, and become better than humans almost instantly. This was just the beginning of the world we're currently in, where we have seen computers essentially in these models exceed human intelligence in a number of different ways. Of course, there was the AlphaGo, also from DeepMind, applying the same reinforcement learning, not just to chess as it had done, where it caught up on IBM's earlier win over Garry Kasparov, but this much more complex game that where rules couldn't be programmed in just because of the almost infinite nature of possibilities on a Go board. But learning by reinforcement learning allowed it to play itself millions of times. And you may remember this kind of rather famous victory over, by a mile, the world's best player at the time, Lee Sedol. And then we come up to the current era, and things have begun to move even faster in the last couple of years. You may remember the moment that ChatGPT was released to the world, and they, I think, almost over the course of a month, had 200 million users, the fastest growing user base of any app in all time, including of Facebook and other of those early models. And that takes us kind of up to date for where we are in our run through the history of AI. So the focus here is, where's the potential for medical AI? So I'll talk a little bit about analyzing images and videos. I'll show you some examples of reducing the burden of paperwork in our healthcare systems, very important, helping make difficult diagnoses. I won't talk much about scaling humanity, but I think this is one of the most interesting areas for the application of AI. Not everyone has access to empathy. Not everyone has access to a doctor with specialist expertise. These AIs can help, I truly believe. Also, we know that education is better if there's one-to-one interactions. We know that behavioral change is better with one-to-one interactions. But again, there aren't enough cognitive therapists, there aren't enough training coaches, there aren't enough educators to allow everyone to get one-to-one education or training. AI can likely help there too. And finally, drug development. I'll talk just a little bit about some of the scientific leaning approaches to these models. All right, so this is some work from our group from just a few years ago. At this point, these convolutional neural network models had been applied to chest x-rays, had been applied to CT scans of the chest and the abdomen and various other places. But at that point, no one had actually built a video model. And being a cardiologist and being focused on the only organ that is in constant motion, about 3 billion times during your life, we thought we should build a model for that. And looking at the ejection fraction, which is the amount of blood ejected per beat from the left ventricle, shown here in blue, that is something that is done manually still across the world by millions of individuals on a daily basis. We were able to train an AI to do that better than a human reader and more instantly, and particularly in the form of arrhythmia shown on the far right, it could do it across many more heartbeats than a human could. And I refer you to the paper for more details. Humans will typically take in many modes of data when they see a new patient. So we would see an electrocardiogram, for example. We'd see an echocardiogram. We might have MRIs, and I'll come back to models that can incorporate all of that data. From this work, just again, a handful of years ago, we fed in both ECGs and echocardiograms and asked the model to tell us whether the patient had a Mendelian disease called hypertrophic cardiomyopathy or had a thick heart because they had high blood pressure. And we have obviously had a training data set that we fed in. And then the details are shown here for our LVH fusion model. Pretty good at predicting hypertension, a little less good at predicting hypertrophic cardiomyopathy. In head-to-head comparison, though, our model was already better than its human counterparts, including some humans who had spent 20 years of their career trying to diagnose hypertrophic cardiomyopathy. I was one of those who was beaten by the AI, so humbling us all. We can also ask the AI, like, what are you looking at? What are you using in this video or in this image to help make your decision? And what it found is that it recapitulated 100 years of learning. Here's T-wave inversion in this electrocardiogram at the top, the asymmetric septal hypertrophy of the left ventricle shown in the echocardiogram at the bottom. Large language models, of course, like the kind that ChatGPT and Google's Gemini and Claude and others are based on, it really came from a series of technical advancements that relate to something called attention. I don't have time, I think, today to really go through that in detail, but attention and multiple heads within a network that kind of refers to itself were some of the individual elements of the kernels that are inside large language models that allowed us to move into this current era. And so when you teach these models medicine, or in some cases you don't even have to, you just need to give them enough data, they start to perform better than humans on medical exams. So this was the first paper demonstrating this. At this point, if you look in the graph here, you'll see that over the course of the move from MedPAM to MedPAM2, this particular model went from being at the level of an average medical student to the point of being an expert medical student. So they did the same in the law degree courses and education courses and many others. These are the medical exams. But these models are surpassing us all around. And this might be illustrated better by this example, for those of you who are a little medical in the class or those of you who are not. I give you this example from GPT 3.5. I just got, thinking of myself, I just got out of a 12 hours of air travel and my left calf hurts. I also have a little trouble breathing and I wonder what that could be. So we fed this question into GPT 3.5 and it said, do you have any pre-existing medical conditions? I said no. It said, in that case, I recommend starting with some light stretches, such as the ones below for your left calf and diaphragm. I hope all of you would have a sense that that might not be the answer and maybe we should take a little bit more care. Fortunately, GPT-4 takes a little bit more care. Says, I'm really sorry to hear you're experiencing these symptoms. It's important to take this seriously. This could be a pulmonary embolism. Indeed, that's exactly what it could be. Those are the cardinal symptoms of a pulmonary embolism. And you should seek medical attention immediately. So this is a change from GPT 3.5 to 4, partly reflecting the fact that these models are just doing better. This idea of the factuality of the answer, because remember, these models simply are next token generators. They basically have a probability of the most likely word from their huge training data, and they put that word together. They don't have knowledge, despite the appearance of even a theory of mind that I showed earlier. This is a paper that we contributed to that's published in one of the new journals, New England Journal of Medicine AI, using a very kind of hot topic in AI at the moment, which is called RAG, basically Retrieval Augmented Generative Data. So a combination model that uses both next token prediction to just come up with words, but also references specific facts to bring you back facts. And if you look in the table at the bottom, you'll see that compared to ChatGPT 4 or Bing or Bard, that's the Google version, Almanac, which is our model trained on medical data, was much more factual in its responses as a result of not just doing next token prediction. Also from the same journal, actually, these two papers are the two most cited papers since the start of this journal, but it's pretty early days. It's only been going for a couple of years, was this paper from the Google team that purported to be able to bring in MRIs and ECGs and X-rays and really multimodal data and essentially interpret it from the perspective of medical diagnosis and treatment. And they even turned it into a conversational agent, so an agent that you could essentially have a conversation with the patient. Tested that against primary care doctors and found, as judged by subspecialists, that it was more accurate and more empathetic, something that I mentioned earlier. So one of the questions I promised to answer at the beginning, are we using this in current clinical practice? This is our adult hospital at Stanford, where the patients from these different rooms get a view of the hills. We are using the GPT version that's embedded in our medical record to start to help answer questions that patients send us. So there's a generated reply shown here, for example, as a potential email that you can use to respond to a patient, and then you get to edit it and send it off. Here's another one where the model actually suggests some specific medical therapy that might be possible for the doctor. And again, you get to edit it before sending it back. So this is a use case. This one was actually studied to see if it reduced the amount of time that was spent. In actual fact, interestingly, it did not. If you look at the underlined area, it improved the mental task load and work exhaustion felt by doctors, very important in an era of burnout, but it actually did not save time. So this is a pilot rollout at Stanford in collaboration with both ChatGPT and Microsoft. Another of those is ambient scribes. So you may have been to the doctor and they may have spent a lot of time typing on a keyboard your notes at the same time, and it's clicking, clacking away in the background. Wouldn't it be better if an AI could just listen to your conversation and write the note for you? Well, it turns out it can. So we have a system called DAX Copilot, again from Microsoft, Nuance, and Epic, that these came from my nurse who sent these to me last night, so hot off the press. You basically turn on your phone, you start the AI system, and it listens to the conversation you have with the patient, and then literally populates your note, shown here in the Epic system. And here's some of the notes that she did yesterday. So it's actually here today and something that we're absolutely using to try and reduce the burden of paperwork for doctors. In this paper, if you're interested, we discussed quite a lot more about how AI is being used in molecular medicine, specific examples from genomics and immunomics and other elements that are being used in the lab from a pathology perspective. We used a deep variant, which is a variant caller from Google for our whole genome work, and it is easily, for long-read sequencing, the best variant caller that is currently on the market, and it's based on a convolutional neural network. So it basically makes the pileup, if you're familiar with how genomic sequencing works with next generation. You essentially pile up the individual words that are read by the sequencer against the reference, and you basically look at that pileup and read down each vertical. In this case, they turn that picture into literally a picture and have the artificial intelligence organ essentially read out the variant. The top-level evidence always comes from randomized controlled trials. So you may be asking, are there any for AIs in clinical care? There's only one that I'm aware of that actually has clear mortality benefit outcome. So this is from China, so a little closer to here than where I normally stand. And this is an important paper published in Nature Medicine, as I say, to my knowledge, the only so far randomized controlled trial showing a mortality benefit. This is an AI that looks at the ECG of every patient who came in during the trial and basically sent an alert as low risk or high risk to the doctors. That was it. Didn't do anything more sophisticated than that. But that intervention alone allowed the doctors to spend more time focused on the patients that really need it. And we believe, the authors would proclaim, and I think I believe them, that alone was enough to change the outcome at 30, 60, and 90 days as shown in the graph at the bottom right. So pretty stunning effect, would need to be replicated in other places, but certainly pretty interesting approach to the application of AI in clinical care. And then I wanted to give you something pretty up-to-date. We released this paper just 10 days ago, and this is a continuation of our work with the Google team, the conversational agent I mentioned a few moments ago. And in this case, we basically take in the reports from ECGs and echoes and MRIs and the genomics for a patient with Mendelian heart disease and basically have conversations with either general cardiologists or subspecialist cardiologists and eventually also with the patients. But we here have the subspecialists judge the workup of the general cardiologist for these patients and then compare that with the conversational agent from Google. And in this case, we showed that even off the bat, the conversational agent from Google actually outperformed the general cardiologist. When we let them work together, as in discuss the case together, they performed significantly better. You can see in red here, shown at the top. The cardiologist, together with Amy, the conversational agent, massively outperformed the general cardiologist without the agent. And this is just to give you an idea of what that might look like. Here, this is a GP talking to the conversational agent. I have this 69-year-old with some interesting findings. The AI says, happy to help. It's always in a good mood. It's always happy to help. I've reviewed the data. What concerns you? And so the GP can ask specific questions. Well, this echo shows this and that, and I'm a little concerned about that. And the AI responds, and so the conversation continues, and they go back and forth. And this is essentially like a specialist cardiologist available to the general cardiologist or to a GP who would then, at the end, get to the point of having a better ability to judge what was required next. So this is the sort of thing that we're currently rolling out and testing actually in our clinics. When we released this paper about 10 days ago, just talking of other general AI things, and you're not going to be able to hear this since it's an audio clip, so maybe I'll say nothing more about it. But there's actually a tool now that will generate a podcast from your paper. So many of you might listen to podcasts. Usually it's two people talking about something together. We fed the PDF of our paper into this tool, and it basically created a very real-sounding podcast between two people who then discussed the paper. If you're interested in that, if you look either on social media or there's a link on the paper on archive, you can link through to the generative audio of the podcast, and it is quite remarkable. This has just in the last few weeks become kind of a viral tool, notebook LMM. You can see the date, September.
Hey everyone, welcome to another
here it
deep
is.
dive.
The audio did work.
Because today we are going deep into the world of AI.
In medicine.
In medicine, yeah. And how it could change how we think about specialist care.
Yeah.
You know, like how we access it, how effective it is.
This is entirely generated
robot doctors?
by an AI.
Like, is that what
these people do not exist?
Because that is what I'm imagining. You know, like in those movies where it's like all these like, you know, blinking lights and stuff. Anyway.
Well, not quite. It's more nuanced than that,
Okay.
but also really exciting.
So you can hear how they talk to each other, you can hear the breathing, you can hear the is and as in between the words. It sounds incredibly real, but these two people do not exist. And so now you can basically do this with anything you have. You can do it with your entire notes from your 4-year undergraduate course, from your high school. You can do it with our paper. You can do it with all the papers you can't be bothered reading. It'll turn it into a podcast, and you can just listen to the podcast. So then in the last 5 minutes, I was going to talk less about the application of medicine and a little bit about some of the ways we're also using these models in science. And this is a paper that came out yesterday. This is not from us, but I had to add it in since we're talking about things that are very up to date. This is from Cell, published yesterday. I've not read every word of the paper yet, but I did read the figures and the abstract and some of the other elements before putting it in. It's basically from Maria Zidnik at Harvard, and demonstrating how we could use AIs at all levels of scientific discovery, from brainstorming about an idea to thinking about causality, thinking about experimental planning. At the moment, the AI doesn't actually do the experiment for you. You've still got to do that. But then addressing the data, of course, analyzing the data, talking to you about the data, displaying the data, and eventually helping generate the paper. It's a pretty interesting read, and I think a way of which, as you can see from our work in the medicine field, the partnership between the human and the AI is, I think, going to be the most interesting thing. But I mentioned AlphaFold at the beginning. Of course, this has been a revolution in our ability to model proteins, and then in the most recent version, AlphaFold 3, our ability to model proteins and their interactions with other molecular entities, so like ions and nucleotides and other molecular entities. You can see some of them modeled there with the pictures on the right. A much harder problem, not as if protein folding wasn't a hard enough problem. And so this is pretty interesting, and I didn't manage to include a slide that one of my postdocs is working on, where we can basically model specific both mutated and non-mutated versions of the protein and their interactions, even with things like calcium ions, when I'm focused on the activation of the cardiomyocytes in the heart. So AlphaFold 3 and protein models in general, a big part of the future of drug discovery, and only getting better. Our most recent work has been in collaboration with this team based out of Stanford and the Arc Institute, thinking about scaling to DNA models. If we think about these language models and what they do, then we want them also to learn the language of life. So we know they can learn proteins. We've seen that with the Nobel Prize. It's a bit different when we think about DNA, and we heard this beautiful talk just a few moments ago about epigenetics. Think about the long-range effects of one piece of the genome on another and how it's all tightly compacted inside that nucleus. In order to do that with a language model, we need long context. We need to be able to understand the relationship between this word here and a word maybe a million base pairs away. And to do that actually cannot be done with the current models. So we've been working with these groups to try and think about long context modeling. The current model is called EVO. There's another model coming up. One of the applications of that would be to think about genome engineering and trying to help solve the delivery problem that we have. We have these great tools, CRISPR and base editing and prime editing, and we want to be able to get those editors to the right cell at the right place at the right time, but we're limited in how we do that. We've used adeno-associated viruses, but they have limited packaging capacity, and some of them, sure, go to the liver, but you try and get them to the brain and not the liver, or the heart and not the liver, that can be much harder. Maybe these models can help by designing new AAVs that are fine-tuned with data that we generate in our own lab, for example, from this human cardiac slice culture. And then we train the model with that knowledge, and then we ask it again to generate new capsid designs that will help us even more to emphasize the delivery to the heart and less to the liver. So just a quick example of that, there's also been novel CRISPRs that have been designed by some companies using a very similar approach. So I think the future looks quite bright for the possibility of applying these language models to the language of life. So in summary then, I have a few questions for us to think about. As you understand, I hope you would agree that we are really in the middle of a revolution that will affect all our lives as scientists and as doctors and beyond. Many of us are now using these models in place of search functionality for Google. They just give better answers. But there are some caveats to think about. Some of them are shown here. There are others. I know we'll discuss probably a few more on the panel later, so I won't go back to that. But I'll leave you just with this last bullet point and the question of safety in medical application of these because of people having access to specialist care. Is an imperfect tool in the hands of many, many people around the world better than a perfect tool in the hands of only a few? I've shown you the work of many people. These are a few of them, some of the people from my lab in my backyard, back garden, as I used to call it, backyard, as I now call it, I suppose. This is part of the team and some of our younger members of the lab shown at the front. Thanks again for the invitation. It was really a pleasure to be here, and I look forward to discussing later.
Thank you so much, Professor Elsley. Please be seated. Thank you very much. And next, we're happy to introduce with you Professor Catherine Shah. As this session's convener, Professor Shah is Emeritus Chair of the Biochemistry and Professor at the University of Hong Kong, member European Molecular Biology Organization, member Hong Kong Academy of Sciences, fellow World Academy of Sciences, member Program Committee of the Future Science Prize Week 2024. Let's give big hands to Professor Shah.
Thank you very much. I am really thrilled to be here and to be part of organizing this wonderful session in life science. We've heard already two absolutely brilliant talks from very basic developmental biology and underlying principles behind epigenetic and genetic inheritance to mind-blowing ideas about how AI is going to change medicine and our daily lives in the future. So the feast goes on in the following two talks, and the next talk is going to be delivered by Professor Lorenz Studer. And Professor Studer is a wonderful example of somebody who has achieved so much as a basic scientist and has gone all the way to clinical applications. Professor Studer is the director of the Center for Stem Cell Biology at the Sloan Memorial Sloan Kettering Cancer Center in New York City. He's a real pioneer in developing methods to differentiate human stem cells to neurons that can be used to treat neurodegenerative disorders such as Parkinson's disease. And he is also the one responsible for having a first-in-human clinical trial taking off-the-shelf stem cells, stem cell-derived neurons to treat a patient with advanced Parkinson's disease. So he has a wonderful background and wonderful research going on about how you can control differentiation of stem cells, not only to understand disease mechanisms, but for their application. So we look forward very much to his talk entitled Directing Cell Fate Developmental Time and Brain Repair Using Human Stem Cells. Welcome, Professor Studer.
Great. Thanks so much, Cassie, for this very kind introduction and for the invitation to be here. It's a pleasure to follow these beautiful talks about epigenetics and about AI with a talk about human pluripotent stem cells. And obviously notice that also the Future Science Prize winner of this year is Honghui Deng, who did beautiful work on the mechanisms of how you can actually reprogram cells back to pluripotent states using just chemicals. In fact, all the work that I'm going to show you has to do with human pluripotent stem cells. And before we get started, I just need to quickly, it moves forward on this screen, but not on this screen. Oh, no, it's coming, let's go one more back. So just quickly my disclosures. And at least one of the disclosures, Blue Rock Therapeutics, which is a company that sponsors now some of the clinical work that I'm going to show you some examples at the end of my talk. Now, really, one of my major goals when I started off my career was really understanding how do you go from a pluripotent stem cells into all the cells of the human body. And that was really kind of a quest that might take decades to really figure out. But by now, at least for the nervous system, I think we have a very good understanding how to do that. So even if you give me any cell type of the brain or the PNS, I've never really tried to generate. I could use the same principles to get very close to generate those cells at high efficiency. And the way it's done is basically 3 major steps. And all those steps follow developmental biology. So step number 1 is you need to decide with regard to germ layer and subgerm layer identity. And so the amazing thing is actually to go from a pluripotent cell to a central nervous system cell is remarkably simple. So it turns it's actually a default in their development. So if you block all the signaling pathways, such as BMP and TGF-beta signaling, they spontaneously convert at nearly 100% efficiency into neural cells. If you don't play with just 2 more signaling pathways, you can now make all the major ectodermal lineages, not only the CNS, which is the brain and the spinal cord, but also the peripheral nervous system, the sensory structures, such as, for example, in your nose or in your inner ear, or also the skin. And so that gives you the first building block to specify those 4 major subectodermal lineages. But step number 2 is actually that for each of those tissues, there is something like a zip code. So it turns out the front of the brain, the forebrain, develops into very similar, very different structures than, for example, the spinal cord. And so what you need to instruct in a second step is not only making the right layer, but the right position in the body. And that's done by using so-called morphogens that pattern cells to those posterior fates, particularly so-called Wnt signals, and all the signals critical for that in an anterior-posterior, from the front to the back, and from the middle to the side dorsoventral position. So that's step number 2. And once you have the right tissue, the right position, the right zip code, you can predict which cell is going to come out from there. Then the final 3rd step is really understanding timing of the process. Because in each of those regions, you actually have a sequence of events that happens from those cells born in this region. They make first neuron type number 1, for example, in the cortex deep layer neurons, then they make upper layer neurons, then they make glial cells, and you can control the timing of that process with certain transcriptional networks, and you can force the cells to exit at step 1, 2, or 3. And so again, if you combine all those steps, you can do that either in 2D, making a layer of uniform cells, or you can also make that quite effectively now in 3D to make organoids in such a manner and to really build kind of an atlas of the nervous system. Many of you heard maybe of the idea of making a cell atlas using single-cell techniques, but the way we understand cell atlas is not only to profile the cells descriptively, but to understand enough that we can actually generate them. And these are just one of those examples. By now, we can make more than 50, it's actually more than 60 cell types routinely in a dish at high purity, at high efficiency, and use then the cells either for modeling a disease or, for example, using them as a tool for cell therapy. And so we really got good at this step, putting those 3 steps together into a logical manner. In fact, one thing is talking about AI is now saying, can we make that even more systematically and understanding basically even cell types we haven't really fully generated with that logic. Now, what we are not as good at is understanding time of the process. So it turns out when you go through those steps, they follow pretty much the human timing. And we all know to make a baby takes you 9 months. To then get to an adult stage takes about another 20 years. And to actually get age-related diseases, often they occur only when you're 50 or 60 years of age. And so this timeline is not really absolutely set in space, because we know in animals they can go through the same steps, but they can do it significantly faster. It turns out the specific factors, the signaling pathways, transcription factors that are important for those cell fate specifications are largely the same. But the way the movie runs, how quickly you go through the program, is actually very different across species. And so this is still one of the major mysteries that is a challenge in the field, because we want to use those cells to study adult or aged diseases. So we need to move the time clock forward to get to that late stage. So we worked on that also for quite a while, and we made some progress in the first step. How can you accelerate the time to go to initial fate? Can you go faster than nature? And we had such examples where we can screen for combinations of small molecules that can do the job slightly faster than normal development to make, for example, peripheral cortical neurons or glial cells in the brain. It still hasn't really explained with those mechanisms how we actually understand species-specific differences. And there's a number of groups that now have looked at that and found that there are actually species-specific differences between a mouse and a human in very fundamental processes such as protein degradation, transcription elongation, other biochemical reaction time or metabolic rates. And in some cases, they seem to roughly mimic the difference in timing between the developmental speed that that cells go through these processes. Now, right now, most of that is still correlation and not causation, because it's very difficult to change protein degradation rate in a systematic way without really messing up whole development. Now, where actually the problem is even more acute is that we have not only a 2 to 3 time difference in going from pluripotency to a cell fate, but a much larger difference between a mouse and a human to go to a fully mature cell. So what do I mean by maturation? Maturation, I mean once we already specify the fate, you make, for example, a nerve cell in your cortex, for the cell to become fully mature takes a very long time in a human compared to the mouse, at least a 10-fold difference. And so the question is, how do we understand that? And how do we model that? On the one hand, we know that it's a largely what we call cell-autonomous process, because you can take a human or a mouse cell, or in this case, a mouse, a pig, or a human cell, and graft them into an animal that has a depletion of that cell. And so now you're going to see over time, the grafted cells can take over the function that's lost in this animal model. And what you find is that the human cell takes much, much longer than the equivalent mouse cell. These are so-called dopamine neurons in a model of Parkinson's disease. So even though they're in the exact same environment, they have a species-specific difference in their maturation rate to become mature enough to cure this animal from the dopamine deficit. And there's other work from, for example, Pierre van der Hegen, where he grafted mouse or human cortical neurons in a mouse cortex. And the human cell takes about 9 months for them to develop in these beautiful pyramidal neurons that if you show them to a pathologist, they tell you, okay, they look about like a neonatal cortical neuron. So meaning even though they are in a mouse brain that develops much faster, they stick to their human speed. And so there's a little bit of work, again, that suggests that, for example, the energy metabolism might be involved in that. So again, work from Pierre van der Hegen showed that if you enhance mitochondrial maturation rates, you can somewhat speed up the process, but it's only a very partial success. What we try to do is, one approach is just simply, can we screen for factors that can accelerate it? So in this case, we developed a chemical screen where you first make the neuron very, very precisely. All neurons are born at the same time. Then you expose them to a large number of chemicals. You wash out the chemicals, grow the cells for another week, and you ask yourself after an additional week without any such treatment, do they remember the previous treatment? And they found, in fact, that 4 such molecules could do that in combination we refer to as a cocktail called gin tonic. You can imagine the student liked to have gin tonic drinks, but it comes really from the letters of those molecules. Now, the way it works seems to be that you actually have to inhibit, we talked about epigenetics before, something which we are referring to as an epigenetic barrier that in human actually resolves extremely slowly to allow maturation to occur at the right speed. And then you have a second set of activity-dependent programs that come in neurons. And once they basically become fully active, trigger activation-dependent maturation. That's, for example, the maturation program that you have in a baby once it's born. You start seeing, you're opening the eye, you get visual input, and you trigger this activity-dependent maturation program. But this cannot really occur if the epigenetic state is not ready to do so. And you can do that both, again, in a 2D model system, or you can do it also in 3D organoids, where you can now get significantly more synapses formed much, much earlier with this gene tonic cocktail, and you get much more early spontaneous activity. Now, when you look at that more systematically, what is really fundamentally happen as the molecular substrate of maturation, Gabriele Ciceri in the lab is a postdoc who really established the atlas of maturation at the level of function, at the level of molecular states, to then map, for example, what happens transcriptionally, moving from a very early pluripotent cell all the way to a young neuron to a more mature neuron, and how can that reflect the maturation that happens normally in the brain, and how we can stage them with regard to their age. We can then look and define the transcriptional programs that are underlying that by looking at the chromatin in those cells. But what maybe was most intriguing is what we found is actually the gene regulatory network that very, very slowly decreases during maturation. And it turns out that most of those factors were, again, these chromatin-related barrier factors that were most highly enriched. And in fact, we found more than 30 such factors that are expressed in young neurons when they are born that very, very slowly get downregulated over a period of many, many months. And it turns out about half of them, if we just simply overexpress those, we can achieve an acceleration either in marker expression or in the activity of those neurons. And what's quite remarkable for those factors is that nearly all of them seem to be retained in individual neurons. So what I show you here is a single-cell data set from the developing mouse brain. And we basically use a tool to just look at gene expression once the cell is becoming a neuron. So this is purely maturation process at this stage. We look again at our maturation factor that came out of our atlas, and we found in all those neurons, independent which neuron they are, this barrier has to go down over time to allow for the maturation to occur. When we then look where do those factors come from in development, we find that they actually express even much earlier than when the cell becomes a neuron. And we can actually now go ahead and inhibit those factors already at the stage when the cell is still a precursor cell, just at the transition to become a neuron. So in this case, you inhibit those epigenetic regulators such as PRC2, which is a polycomb-related epigenetic regulator. We can inhibit those just transiently in the neuroprecursor stage, take away those chemicals, let the cells allow into neurons, and now the neurons mature more quickly. That's shown with this maturation scoring factor. For cells, again, they were treated over here outside of the graph, but the cells remembered that treatment and now mature more quickly, a little bit more akin what you see in a faster developing species, such as in a mouse. You can, again, measure that in many different levels. You can look at excitability, synaptic connectivity, activity in those organoids, for example, again, can be done in 2D and 3D, and all those factors respond in a similar way to now show that just simply by inhibiting those transiently this barrier, we can get a faster maturation occurring that seems to persist. Now, I don't go too much into the mechanism, because obviously it's a quite complex underlying mechanism with regard to the factors that contribute to this epigenetic barrier. One of the most powerful factors among those was, again, factors that inhibit this PRC2 complex, these EZH2 inhibitors. And so we then asked mechanistically, so what does that mean for maturation? We had a beautiful introduction about epigenetics, about DNA methylation, but also about histone modifications. And PRC2 is particularly important for one of those histone modifications called HDK27 trimethylation, which is an inhibitory such modification. So what we did here is we just simply looked in young neurons, or in more mature neurons, or in precursor in neurons, what distinguishes basically this chromatin signature in the cells, and which of those chromatin patterns on a set of genes, so this is a set of genes that has this specific pattern, another set of genes that is deposited chromatin with this specific pattern, and so forth. So you want to know which of those patterns actually correlated with maturation. That's shown here. So these are genes here that show this maturation-dependent expression pattern, so they increase. And we found that these are particular genes that have a so-called bivalent state, where they are ready for activation, but they don't really show you gene expression. And that seems to be one of those barriers that needs to be removed when a neuron wants to fully mature at a certain speed. And so we showed that indeed we can then inhibit those PRC2 component factors with CTH2, inhibit those specific inhibitory factors, and show that indeed we, for example, affect this deposition of HDK27 at the genes important for the maturation process. So the summary of this part is then really that we have now, I think for the first time, a good way to understand why do human cells go so slow. And maybe, if you say, why has nature designed it like that, it's maybe quite obvious. The brain is extremely complex. You have 100 billion neurons, and they have to be all born over a certain amount of time. They have to connect with each other under those conditions, and it doesn't make sense now to make them super fast before they are even ready to properly connect. So it's like a symphony of factors that has to go in the right sequence and events to actually achieve the enormous complexity of the human brain. And again, we have now at least some glimpses of how that works, how we can inhibit this barrier either in the neurons or already in the precursors. And we know other components that contribute what's called in gene regulatory network is epistatic interactions of what you can actually inhibit and what is required for the inhibition to trigger a maturation program. Now, coming back to the mouse and the human, if you actually look, so is this program in any way different in a mouse or in a human? It's clearly not going to be the only difference between species, but we can actually use methods or single molecule counting of component of the epigenetic barrier, shown here, like ECH2. And what we find is actually that the human has at the equivalent stage is a higher barrier than the mouse. So this is a very primitive way to show it, but might in part explain again why it's much more difficult to get this barrier to decrease over time in a human cell than in a mouse cell. And conversely, you can now actually do the opposite, and basically with that knowledge engineer mouse cells that now, for example, develop slower, more human-like. And so this gives you again kind of a glimpse of some of the changes that we can use to control timing in our human stem cell system. And there are many other projects ongoing, for example, in the lab. For example, one idea that's even more radical is that you can now fuse human cells with mouse neurons and see, do they now go at the speed of a mouse or at the speed of a human, which was actually a very critical experiment pre-Yamanaka, figuring out where all the factors that can induce pluripotency are present in an embryonic stem cell. And in this case, we actually have now evidence that indeed we can accelerate human timing in these hybrid neurons. And we have also another story, which maybe soon comes out, where we did genome-wide CRISPR screen and again came across this epigenetic barrier as a key component of the timing factor. And then the final part really of the timing question is, we don't really just want to make mature neurons, but we also want to understand aging, what controls aging, and maybe what we can do to reverse it eventually. And so in this case, we have just recently published some of the criteria how you, at the cellular level, do you actually know that you have an aged cell or you have a young cell. And these are some of the key factors that distinguish those cells, and some of the manipulations that we and others have published previously try to induce an age-like state. And just very recently, we've performed a genetic screen that found a new player in this pathway, which is called protein nedylation. It's a pathway that's very important, interacting with another protein degradation pathway in boosting degradation of proteins at a certain time point. I mentioned you protein degradation is very important for timing. And in this case, we can actually just add a drug transiently to certain neurons that have a mutation in either Alzheimer's gene or in a Parkinson's gene. And if you do that in a young neuron, you can measure the changes related to that mutation. For example, they have more of this Alzheimer protein called A-beta, but they don't degenerate. But if you now do the second step, you inhibit the NADylation, you trigger an aging program in the cell, and you can actually, for the first time now, get reliably see the tau phenotypes and the degeneration phenotype in those systems. And it works beautifully for both modeling Alzheimer's disease, which was shown in this study, but also actually for modeling Parkinson's disease, where you get only in a genetic context the combination of the genetic vulnerability and the age of the cell converge into triggering and modeling degeneration. Now, having talked about Parkinson's disease, that's really then the other approach how you can use stem cells, not only making the right cell, the right genetic modification to model the disease, but using the cells for the actual therapy. And so this is an attractive approach in Parkinson's, because it turns out all the classic motor symptoms of Parkinson's disease are caused by the loss of a very specific population called midbrain dopamine neurons. And I mentioned to you before that the brain, human brain, is about 100 billion neurons, but it turns out dopamine neurons are about only 300 to 400,000. So it's a very rare population of cells. And once you lose about half of those, you start getting symptoms of the disease. So therefore was the idea, if you know that those cells are lost, they are the cause of the symptoms, can you just simply put a few 100,000 cells back and rescue those symptoms? Now it's important to understand that Parkinson's is not just a dopamine neuron disease. There are other symptoms as well, for example, problems in sleep, in the gastrointestinal tract, in the context of smell, and even at later stages, cognitive dysfunction. And so those symptoms are likely not going to be affected by dopamine grafting. So what currently we try to do in a most positive way, we try to cure the movement disorder of Parkinson's disease, but we might not truly cure Parkinson's disease with regard to the other symptoms. Now, this idea has been around for a long time. It was actually done first, even when I was back in Europe, very involved in some of those trials, using fetal transplantation, where dopamine neurons were plucked out of a developing fetus and then grafted into patients. And what we learned from those studies is that the cells can survive in a Parkinson's brain. They can actually survive for up to 25 years, which is quite remarkable, 24 years without any immunosuppression. The problem really with fetal tissue was it was very difficult to procure, highly variable, and placebo-controlled studies didn't really show a consistent effect, in part because it was so difficult to design consistent trials. Nevertheless, some of those patients were followed for more than 15 years, and some of them, admittedly a small subset of them, showed remarkable effects, meaning they actually had, for example, completely could stop their L-DOPA therapy, which is the most common therapy that these patients currently get. So this was quite remarkable and suggests that this could work, but on the other hand, it might never really work properly with fetal tissue. And this is a paper that's soon going to come out from our colleague of ours, Roger Parker, that went actually back and tried with our knowledge now, 20 or 30 years before these initial studies were done, can we go back to fetal tissue and do it better? And as you will see, if you read this paper coming out soon, there's still a lot of problems with that. Even with today's knowledge, fetal tissue is too variable, too difficult to procure and standardize. And so that was really my vision when I started actually going to the US at the time, that we need to have a stem cell source to really achieve that. And it took us about 10 years of work to figuring out the developmental biology to make the right dopamine neurons. I'm not going to go into details of that, but it's a cell that can survive in a mouse, rat, or monkey model of Parkinson's disease. These are models, very simple ones, like this rotation model. This is obviously a sped-up movie, but it's an animal that's lesion on one side of the brain, has no dopamine on one side, and once it's grafted, it becomes symmetric. There's another animal cannot really properly initiate movement with the left paw. It's not paralyzed. It's not like you have a spinal cord injury. It can move it, but it cannot initiate movement, which is exactly what happens for a Parkinson's patient. When, for example, a Parkinson's patient wants to move across the street, the light turns green. They always have a long time before they can actually initiate movements. Then they have shuffling movements to move forward. So that's what happens here. So the animal drags the paw, but then 2 or 3 months later, he can again put weight and properly move the paw. When we graft our dopamine neurons, we also know exactly how those neurons work. So it's not just a black box, putting them in and the animal goes better, but you can actually do so-called optogenetic studies, where we take the same dopamine neurons, but we put in like a light switch. So you can now graft the neurons, but you can now shine light on the brain, and when you shine light, just during that time, the nerve cell shuts off. And if you do that, within a minute, all the benefit you have from the animal, from the grafting, goes away. We can also show that once the activity goes away, that it's activity dopamine release that causes the benefit. And what's shown here is even more sophisticated. That's what the neuroscientists do. They actually stimulate certain nerve cells in the region of the brain, the cortex and thalamus. They record from these blue and red neurons in the striatum. And these are the neurons, the circuit that's modulated by dopamine. And you can show exactly when you switch on the light, that the strength of the synapse drops. When the light is off, it goes back to the normal strength. So what that means is these dopamine neurons work exactly the way normal dopamine neurons are supposed to do. With these basic insights, we then worked for nearly another 10 years, getting all the regulatory approvals to move these cells to the clinic. We had to do extensive preclinical studies in hundreds of animals, in rats and in monkeys. I'm not going to bore you with those studies, but just one example to show you for the efficacy, how it works, is you have this lesioned animal, so lost dopamine on one side of the brain, and after grafting, you see it's, again, pretty much fully symmetric. And this rotation movement that I showed you before in the video, from 10 rotation, goes back to 0. So this led then in 2011 to the first time that we were allowed to use these off-the-shelf dopamine neurons. We made billions of those neurons in hundreds of vials to be ready for grafting. We did that in New York in MRI-guided OR, where you can very precisely deliver the cells at the right site. You do this implantation at 3 different tracks, 3 different deposits in each track on both sides. And you can then see actually the tracks right after grafting. So this trial basically involved 12 patients. They are now all grafted, and they're all at least 2 years after initial treatment. 5 patients were at the lower dose, and 7 patients at the higher dose of treatment. And we had also exclusion criteria. Again, if these patients have, for example, dementia or other issues, they were not included. Now, this is a phase 1 study, so the most important thing you have to report is safety. And indeed, we had excellent safety, so we had no therapy-related adverse events. And we had also no of the side effects that were observed with some of the fetal grafting in the past, because again, we can much more precisely shape the cells and avoid the cells that we think caused the graft-induced dyskinesia in the past. We then can show that the graft actually survives and functions. So this so-called PET imaging, where you have a dopamine tracer, this is pre-treatment. And you can say usually this dopamine signal, that's a PD patient, gets weaker every year. But in fact, after transplantation bilaterally, it gets stronger over time. And it gets stronger even after the time we, for example, remove immunosuppression in those patients. And again, I don't have time to go through all the clinical scores. I'm just going to show you one example of a clinical score now at 2 years of time, which is the so-called UPDRS score. This is a score where patients are rated by motor function. And once you have about 40 to 50 points, you have pretty severe Parkinson's disease. Every year you look at initial 2 to 3 points, you get worse. And when you get about 5 points difference, that's really clinically meaningful. And what we found is, particularly in the high-dose group, is that the patients drop by more than 20 points. So that's more than 4 times of what's clinically meaningful. And so we were very, very excited. In fact, we saw an effect that maybe was even better than what we dreamed of when we started with regard to that. And we also could avoid some of the side effects. This is what's called the graft-induced dyskinesia. And so therefore, we seem to have a very promising strategy. Now, again, obviously, it's still early stage. But we know that we can deliver the cells safely. This is the first off-the-shelf dopamine product in the field, so it could be scaled potentially to many, many patients. We have evidence that there's a dose dependency. We can avoid some of the side effects that have hindered this approach in the past, but obviously we have only a small number of patients so far. And so what really needs to be done now is to scale that up to a larger study that is starting in 2025, which we hope that could be an actual registration trial. just the last minute, we also think, obviously, what's next? On the one hand, we can still even make these cells even more precise than in the past. So it turns out they're not only dopamine neurons, but the different subtypes of dopamine neurons. And we have now very good understanding, much better than when we started the clinical trial, of how to make those cells using single-cell approaches, basically in intelligent design, and then making selectively just one versus the other dopamine neuron subtype. We understand better what's the limiting factor in these cells to survive in vivo. And finally, we can also endow these cells with additional factors to either completely avoid even the transient need for immunosuppression, or to protect them from the ongoing disease that might affect these cells, we estimate maybe after 10 or 20 years after grafting. And just for this one, we just had a paper earlier this year to show that we actually now understand what limits the survival of these cells, where we did a CRISPR screen that identified p53 as the culprit of a neuronal death response when we graft. So typically, about 5 to 10% of the neurons actually survive, 90% die. The way we solve that clinically is we just graft more cells. But we obviously want to know what is really the mechanism behind it. And we found that this is actually TNF-alpha that's triggered this response, and we can use commercial TNF-alpha inhibitors to prevent this response. We also know that it's actually graft-derived TNF-alpha, so there's some interesting mechanism of how that intersects and triggers this p53 response. And then finally, we can put all this together and now graft not only our early, relatively first-generation product, which are dopamine precursor young neurons, into making highly precise dopamine neurons and subtypes, and getting those to survive reliably in animal models of disease, to maybe be the second or third generation product that ultimately should help the patients. And last but not least, I want to just quickly mention some of the key people of the work who are important for the maturation work. Emiliano, Andrew, and Gabriele, with the paper just published earlier this year. Natalie, and then the larger team, including my colleagues at Blue Rock, and a partner over many years, Dr. Tebar, who was doing most of the surgery in those 12 patients today. And we're going to stop here and hope we'll have later an interesting discussion. Thanks so much.
Thank you, Lorenz, for a truly inspiring lecture to help us to see the relevance of developmental biology all the way to treatment in patients. We continue our journey now with more exciting talks from Professor Hai-Fan Lin. Hai-Fan is the Eugene Higgins Professor of Cell Biology at Yale University. He's a member of the National Academy of Sciences of the USA and American Academy of Arts and Sciences, and also a foreign member of the Chinese Academy of Medicine, Chinese Academy of Sciences. Hai-Fan is a pioneer in stem cell biology and cell biology, and discovered a very unique category of genes called the Pee-wee genes, which I think he's going to talk about today. And this was a fundamentally really an important discovery, which was cited to be one of the 10 most exciting discoveries of the year when he produces. He's also a very big leader in many organizations, and in particular in stem cells, as the just past president of the International Society for Stem Cell Biology. And today we are in very good company, because Lorenz will be the president-elect in a couple of years' time. So we have a very good representation of stem cell biologists here, and so we welcome Hai-Fan very much for your lecture.
Well, Catherine, thank you very much for your kind introduction. I wish my mother's to hear this introduction. I first of all like to take this opportunity to congratulate all the recipients of this year's Future Prize. The groundbreaking discoveries epitomizes what's called original and impactful research. And as you've heard from Kathy's introduction, I'll be talking about our own discoveries related to stem cells. And as you probably know from Lorenz talk, and we'll be hearing from this year's recipient, Dr. Hong Kwek Deng's talk. Stem cell research represents a very exciting frontier of biomedical research, holds great promise to cure diseases and regenerate organs in our tissues. However, I want to tell you about our own work that actually leads to discovery of a new paradigm of gene expression from our stem cell work. So when you think about stem cells, you often cannot help but relate these two words to longevity and immortality. And this actually, the pursuit of immortality has been a long pursuit in human history ever since the dawn of civilization. For example, more than 2,300 years ago, Emperor Qin, after uniting China, his next pursuit was to pursue a magic potion that will make him live forever. He ordered the alchemist Shifu, who led an impressive fleet of 3,000 young men and women sailing towards the mystic islands in the east, where Shifu claimed to have these magic potions. The fleet disappeared in the ocean, and later a small island country called Japan appeared, but Emperor Qin never got the magic herb. He died at the middle age of 49, and we started stem cell research just a little bit too late to save his life. Today, we know that there's no such a thing called the magic potion that can make you live forever. However, stem cells are perhaps the closest things that we have in real life. Then what are stem cells? So Lorenz already shown you that stem cells can generate neurons, but let me just take one step back by showing you that totally in our bodies, there are two types of stem cells. Embryonic stem cells that exist in our early embryos, they can generate all types of tissues and cells in our bodies. And the second type is called adult stem cells or tissue stem cells. They usually can only generate differentiated cells within their residing tissues. For example, neural stem cells can only give rise to neuron and other accessory cells, but not pancreas or bone, for example. Despite the big difference between these two types of stem cells in their pluripotency versus multipotency, they have a common property, namely they all have the ability to renew themselves. Therefore, these cells are viewed as the fountain of youth that can provide so-called endless supplies to body's tissues, at least during our lifespan. Now, then the question becomes, what makes these cells to have this amazing ability to self-renew? People have been looking for genes that controls their behavior. And more than 20 years ago, my lab discovered a gene family called the Argonaute PV gene family that turned out is essential for stem cell self-renewal in both animal and plant kingdoms. So we started this work by using little fruit flies, Drosophila, as a model. And as you can see, this little fruit fly has a very powerful, been a powerful model for genetic studies for over a century. Many of the important genetic laws and genes that applies to clinical applications today are actually initially discovered in these little flies. So we thought these little flies could be also a great model for stem cell research. We decided to focus on the ovary of the flies because it contains a small number of actively dividing germline stem cells that can produce many, many eggs throughout the lifetime of the fly. So shown here is a normal ovarian unit in the fly. You can see the two germline stem cells here have undergone many self-renewing divisions to produce a long string of developing egg chambers, which are all colored in brown here. Now, if you knock out a gene called PV in Drosophila, then these two stem cells will just self-differentiate into two egg chambers, but no longer can self-renew to produce more egg chambers. So this phenotype showed clearly to us that this PV gene is essential for the self-renewing division of stem cells. And we showed that this function actually is highly conserved throughout the evolution. For example, in mammalian system, if you look into the testes, This is a structure of testes called the seminiferous tubules, which is the site of sperm production. You can see there are a number of stem cells sitting there. By now, in this young mouse, they have produced many differentiating germ cells and eventually many mature sperm. However, if you knock out a PV gene in mouse, in this case called the mouse PV-like, or MILI for short, you can see these stem cells will still be there. Let me just orient you with these stem cell daughter cells and sperm here. But however, these stem cells will be just sitting there and doing nothing. So therefore, this testis tubule that's actually shown in the mutant under the same magnification as in the wild type appear to be much smaller and contain only the inactive stem cells. So then what's the structure of this family of genes? This family of genes actually encodes a protein that contains 4 functional domains: the N-terminal domain, the central PAG and PV domain that together can bind to small RNAs, and also a C-terminal domain that resembles RNase-H. That means this domain may have RNase or even nucleic acid cleavage activity. And here is a 3-dimensional structure of these proteins. As you can see, the PAG and mid-domain together form a cleft that can hold these small RNAs. So we then further divided this protein family into Argonaute and the PV subfamilies. As many of you probably know, that the Argonaute subfamily proteins, they are central engines of the RNAi and microRNA pathways. They bind to siRNA and microRNAs, and they are always about 21 nucleotides long. And both the Argonaute proteins and these small RNAs are expressed in most cell types. Therefore, today, RNAi and microRNA pathways work in most cell types. And I was very delighted to learn that 2 friends of mine, Victor Ambrose and Gary Rufkins, they received the Nobel Prize last month for their work on microRNAs, in addition to the Nobel Prize given to the siRNA work several years ago to Andy Fire and Craig Melrose. My lab have actually been focusing on the PV subfamily because these subfamily proteins are mostly only expressed in the germline and primitive stem cells. About more than 10 years ago, as Kathy mentioned, my lab and a few other labs discovered yet a new class of small RNAs that we named PV interacting RNAs, or pRNA for short, that are bound by these PV subfamily proteins. And just like PV proteins, these RNAs are also expressed specifically only in the germline and stem cells. The next slide will show you a brief comparison of the salient features between the pRNA and microRNAs. As you can see, pRNA is a bit longer than microRNA. In addition, pRNAs are much more complex than microRNAs. So today, my lab has cloned more than 20 million of pRNAs, which is more than 700 times greater than the total known number of genes in our genome, which is in sharp contrast to the about 1,000 species of microRNAs that we discovered so far. Now, in addition to this complexity, these pRNAs are also amazing in that they correspond to all types of genomic sequences, which again is in contrast to microRNAs, which only target the 3' UTR of mature mRNAs. So of course, the biogenesis has to be different. pRNAs are mostly produced from very long single-stranded RNA precursors, in contrast to microRNAs that are produced from hairpin structures. So we were very excited about these discoveries because if you think about the grand scheme for molecular biology research in the past 6 decades, the research has been focused on the central dogma, which tells us that the genetic information DNA is transcribed into messenger RNAs, which are then further translated into the proteins, and that's how life gets started. The discovery of siRNA and microRNA pathways further enriched this dogma by showing you that there are 2 new exciting mechanisms there to control the stability and the translational efficiency of this genetic information. However, if you take a critical approach to look at the central dogma, actually it's quite limited because it only talks about a few % of the genome in mammals. then how about the vast majority of genome that used to be called junk DNA? So I prefer to call them terra incognita because we know so little about these regions in our genome. And what we found is that most of the genes that are encoding the pioneers are actually hiding in these so-called junk DNA region. And this discovery excites us because we feel like if you liken our genome as the world, then the traditional genes are like the old world, yet we suddenly found ourselves landed ashore of a completely new world, now with new type of genes that make new types of RNAs. And this was a pie I baked for my lab to celebrate the discovery of pRNA, Elisa's pRNA. And as you can see from the trembling hand here, you can tell how excited I was when this was found. So, and as Kathy mentioned, this discovery was selected by Science as one of the 10 breakthroughs that year. All these hooplas aside, an essential question remains, so what's the function of these pRNAs? So today I'd like to show you these pRNAs have two important functions. One is when they and the PV proteins are in the nucleus, they play a very important role in epigenetic regulation. And when we talk about epigenetic regulation, I'd like to thank Professor Anne Ferguson Smith, who has articulated so beautifully about what's epigenetic regulation to you. So let me just briefly recap. Epigenetic regulation refers to heritable changes in genetic information without changing the DNA sequence that eventually leads to the programming of cell fate and to create an organism with diverse cell fate and multiple cell types. Epigenetic regulation, obviously, today have been focused on identifying epigenetic factors and illustrating how they modify protein and DNA modification status, just as Anne showed earlier too. Those epigenetic factors can methylate DNA, or in this case, I'm showing some epigenetic factors. They can possibly do other things, like HP1 means heterochromatin protein combined to the histone associated DNA to make that part of the genome become heterochromatin. Now, if you think about these important factors, one question actually begs for answer, which is most of these epigenetic factors, they're actually just enzymes. They do not bind to DNA, let alone have DNA recognition ability. For example, HP1 doesn't really directly bind to DNA, and histone methyltransferase doesn't really directly bind to DNA in a sequence-specific fashion either. So then to me, a central question in epigenetics that need to be answered is how these epigenetic factors are guided to specific sites in the genome in the right type of cells at the right time. For example, guided to here, but not here or here. So today I hope to tell you that PV pRNA pathway actually play a major role in this process, at least in reproductive cells. So when we started our work, a few transcriptional factors were shown to be able to recruit a few epigenetic factors to the promoters of several genes. And that is an exciting first step, but to me, it reflects more an exception than a norm in the genome, because truly there are only a few genes were shown at that time for this kind of guidance. And also, it's a feedback mechanism. So we decided to look into whether PV proteins have an epigenetic role because it was present in the nucleus. And when you do genetic analysis, it shows all kinds of phenotypes that qualify to be an epigenetic factor. And here is a very visually clear cytological analysis to show you that in the Drosophila polyethylene chromosomes, which are standing in red here, PV protein, which is in blue and green here, actually can directly bind to many sites in the genome. Now, if you further this analysis to molecular level, shows the Drosophila genome here with a telomere of X chromosome here and the centromere here, as well as the left and right arms of second chromosome and the left and right arms of third chromosomes here. You can see when you map pRNAs, this is 14,000-ish PV-associated pRNAs that we first discovered in fly. you see these little peaks. Each peak represents an individual pRNA. They are there scattered throughout the entire genome in all kinds of genomic sequences. Especially they are in the pericentromeric heterochromatin, and they're also enriched in telomeric heterochromatin. Of course, they're also there in many euchromatic sites. So our study in the past 10 years or more allowed us to show that these pRNAs and PV proteins together, they can form a formidable guiding mechanism to pull epigenetic factors to many sites in the genome. So using, again, here one particular PV protein called mu in mouse as an example. Now, in mouse germ cells, the pRNA coding gene will produce a very long single-stranded pRNA precursor, which will then be chopped by the mouse PV protein called MUV2, means mouse PV number 2, that's localized in the nucleus. And this protein's nucleus activity will chop the precursors to produce mature pRNAs. And the mature pRNAs will be bound by this MUV2 protein to form many pRNA MUV2 complexes. And these pRNA MUV2 complexes then will target genomes with the complementary sequences. For example, this green pRNA here, we're targeting this particular gene called the RAS-GRF1 that contains a green pRNA complementary sequence, which is colored in green here. And that will lead to the recruitment of the PV pRNA complex specifically to this locus by targeting the nascent transcript that's still tethered to the pol II. And that will then further lead to the recruitment of DNA methylases to this site. And that will lead to the site-specific methylation at this locus. And as you know, that site-specific methylation, again, as Anne has illustrated to you, is a very important form of the epigenetic regulation, and that will contribute to the definition of these cells into the stem cell fate. So now, if you look into this question again, what I hope to have shown you today is that we actually have discovered a major mechanism that can guide epigenetic factor to the target site in the germline genome and in other cell types that have pRNAs. However, when we looked into all the PV proteins, not all PV proteins are in the nucleus. For example, this PV protein in mice called MUV2, means mouse PV, actually is in the cytoplasm. So this is the seminiferous tubule of a juvenile mouse testis, now standing in green for laminin to outline the tubule itself, and the MUV protein is standing red. As you can see, MUV protein is present only in the cytoplasm. Then what's the function of the PV pRNA complex in the cytoplasm? It turned out they have a second very important function, which is to have a post-translational regulation effect throughout the entire genome. To address this question, let's again go back to our genome. Our genome contains 23,000 genes. In addition, there's a vast intergenic region in which many transposons reside. For example, in mice and humans, we have 1 million transposons residing in our genome. As Anne told you, that's about 40% of the entire genome sequence. And 96% of these transposons, they're actually Rachel transposons, they make intermediate RNAs in order to jump. And in addition, more recently, my lab and others have found that there are many other genes also in the genome, in addition to the protein coding genes and transposons, which are mostly viewed still as selfish parasites that invaded our genome that we had to cope with. Now, these other new genes encoding the linker RNA coding genes that produce tens of thousands of linker RNAs, and the pRNA coding genes that produce tens of millions of pRNAs. We should not forget that our genome also contains a large number of pseudogenes. For example, the human genome contains 35,000 pseudogenes. Most of those pseudogenes can be transcribed into RNAs, but because the open reading frame is mutated, these RNAs cannot be further translated into proteins. And these pseudogenes are often viewed as a defunct carcass of our active genes on their way out during evolution. Now, despite the conspicuous presence of these genomic elements, we still should remember in our genome there are sequences which are important structurally for cell division and for chromatin maintenance, namely centromeres and telomeres. So in the past couple of years, my lab discovered that even centromeres are made into RNAs. They're called satellite RNAs. Now, we call all these different types of genes and sequencing genome as different constituents. So our genome is not just made of 23,000 genes, but millions of these kind of different constituents. And despite their presence, very little is known about their functional or regulatory relationship. So in the past few years, we've shown that actually the transposable element can play a very active role to regulate the expression of many protein coding genes at the post-translational level. In addition, we showed that pseudogenes can also regulate the expression of the cognate sister genes. Furthermore, we showed that transposons can also regulate the expression of many linker RNAs. And now, most recently, we showed that all these regulations are mediated by pRNA pathway, including the functional pRNA pathway in regulating the centromere function during cell division by regulating their product RNA, namely satellite RNAs. So how are these regulations achieved? It's actually achieved through this process. Let's go back to the central dogma again. So the central dogma tells us that a gene is transcribed into messenger RNA in its precursor form, which is then capped and tailed and then spliced to become mature messenger RNA, and then got exported into the cytoplasm. However, we found that in germ cells and many other cell types, transposons are actually also expressed, even though at a low level. And these transposon pro-RNAs will be processed into pRNAs, which are then bound by PV proteins to form PV pRNA complex, which will then allow this complex to target those messenger RNAs which contain transposable elements in the 3' UTR of their sequences. You feel like many people have studied our transcriptome, and tens of thousands of labs have looked into messenger RNAs closely. But it's interesting that very few reports, if any, have reported that messenger RNAs actually contain transposable elements. And we actually found that in the mouse genome, 37% of genes have their messenger RNAs containing transposable elements. So just for young scientists in the audience, this study taught me a big lesson. Even if you're doing genomic study, you still follow the conventional wisdom that cell biologists often say that you only find what you are looking for. You have to ask the right question. And so we found that come to human, amazingly, the number of genes that contain transposable element in the 3' UTR now increased to 50%. Means 1 out of every 2 genes in our genome actually has transposable element bits and pieces in the 3' UTR. We have no idea how these elements get into the protein coding genes, but we do know they play a very important function. And now we should be also cognizant of the fact that the pseudogenes are often transcribed, and they will make RNAs that will be further processed into pRNAs, and the pRNA will be bound by PV proteins to form this PV pRNA complex containing pseudogene antisense pRNAs that will be against the sister gene throughout its entire length. And this is how, in reality, in the most important type of cells, germ cells in our body, how gene regulation is happening, not just the central dogma. And this also is true for the regulation of non-coding RNAs. When linker RNAs are expressed, we found that at least 30% of linker RNAs contain transposable elements. And these linker RNAs then become the subject of regulation for these pRNAs that are regulated by, produced by transposons. And through that, they actively regulate the linker RNA pathway. So just to summarize my talk today, I've shown you that, first of all, if you liken our genome as a world, in the old world, we should not only remember that we have 23,000 traditional genes. Our old world also contains many pseudogenes and many transposons. And now many labs, including my labs, our work have shown that there's a completely new world that exist in our genome, and this world contains many genes that encoding for piRNAs, for linker RNAs, and for other type of non-coding RNAs. And recent work in my lab has shown that these two worlds are actually closely interacted and closely connected. For example, piRNA genes can regulate the expression of traditional genes. piRNA genes can regulate the expression of linker RNAs, and even the transposons can regulate the expression of traditional genes, and even the pseudogenes can regulate the expression of the assisted active genes. And finally, piRNA pathway can even regulate the function of centromere and telomeres to maintain the genome integrity during cell division. So I'd like to acknowledge many former and current lab members who contribute greatly to the discovery that I covered in my talk, and especially these three individuals, Hang Yin, Toshi Watanabe, and Niels Nicholson. They made major contributions to the discoveries I talked today. I'd like to stop here, and thank you very much for your attention.
Thank you so much, Professor Lin. Please stay with us. And could you please proceed to another side of the stage? Thank you very much. Yeah, I can stay with you. Yeah. Maybe we can stay another way. Then they can have more, for the arrangement. And just in our keynote session, speakers explore the groundbreaking research shaping the future of human biology and health. Topics include the significance of epigenetics, AI's role in biomedical advancement, stem cell applications for brain repair and genetic regulation via the PV-Pine RNA pathway. While we are doing the preparation, you may find a robotic arm just over there at the foyer area. Actually, it's for creating the Chinese traditional ink style portrait through the AI technology. So let's have a try today or tomorrow. And then now we're going to move to the panel discussion. May I invite our moderators, Professor Tony Shikam-Mok, and also Professor Catherine Shea, and our panelists, including Professor Euan Ashley, Professor Anne Ferguson Smith, Professor Lin Haifan, and also Professor Lorenz Judah. So I may pass the stage to our moderators. Thank you.
Thank you so much. With such an excellent talk, I'm sure we got a lot of questions. Why don't I kick off with one? And then if anyone among the audience we also would like to entertain your question as well. I start with a tough one, Euan, an AI question. Now, you just mentioned that AI may have a mind, but when there's a mind, the mind can lie, or the mind can learn the wrong thing if the data is wrong. So when I get something generated from AI, how can I know if they're lying to us, whether it's the correct data? How can we deal with this information? In a sense, is that how much can we trust the AI?
Yeah, it's a really great question. I mean, I think that the veracity of data is an issue wherever it comes from. And so wherever on the internet you find it, even if it's a Google search or a Bard search or Baidu search, then you still have to decide whether you trust the data. So wherever the data comes from, you need to apply your human brain towards it. It's a bigger challenge with language models because essentially, as we discussed a little bit ago, they're next token predictors. They basically are making a probability guess as the most likely word from all of their training data that would come next. So they're not directly connected to truth or to knowledge in that way. But you can directly connect them to knowledge or truth in a couple of different ways. One I discussed on our slide, this RAG model approach where you take retrieval augmentation. So as well as next token predictions, you also can have the model go to the internet or to some other trusted source and pull back specific data, specific facts, and have it surface that. And they've been shown time and again, that approach has been shown time and again to be more accurate and to help with this hallucination issue. I think the other is that there are certain questions that you could just imagine a next token prediction will never it's just not equipped to answer. For example, how far is it from Beijing to Tokyo? A language model isn't going to give you an accurate answer to that because you need a model of the world and it needs to be geographical. But there are plugins, for example, there's one from Wolfram Alpha that you can go to that does have a map of the world and that is able to measure things in it. So again, I think we're augmenting the language model prediction with facts about the world. So that's some part of an answer. I think hopefully the example I gave from GPT 3.5 to 4, where it was essentially not recognizing an important medical condition, and then the next version did recognize and give the appropriate advice, I think should give us all hope that we are making progress, but it's still a really important point, and it's not fully solved by any means. Thank you.
So would the others like to make a comment about AI?
Well, I am a very positive person, and I feel AI definitely is, like a flood flooding into our field, but it's a good flood, and we are opening our arms to embrace this. And in my lab, we also have studied AI approach, but definitely not to your level. We are beginners.
Yeah. Can I ask a question actually about the difference? You used that really nice example of the deep vein thrombosis, which is a very striking example. What was it between the two algorithms that made that difference to go from being quite wrong to being more correct?
Yeah, so they haven't I mean, of course, there are lots of examples like it. The difference between 3.5 and 4, as it turns out, although it doesn't sound like much, because 3.5 sounds like, halfway between 3 and 4. But the reality is that GPT-4 is a vastly larger model, first of all. And the basic way these models are put together is that they're trained on large corpuses of data. So there's a lot more data in it. But then they're also, the reason that they pass the Turing test, I mentioned Alan Turing, and can fool us into sort of believing they're human or being more empathetic than humans, is because of other elements in which they're basically trained by humans to react more humanly. So reinforcement learning, like the Atari game, is applied so that because when you start these models and initiate them, the answers they give aren't very human-like, and sometimes, if the data input is small enough, they don't even look like words. And as the data gets bigger, it starts to look like words. To get it to sound human, you have to tell it what's human-like. And so they get trained reinforcement learning by humans. Another example comes to mind is that certain words have become much more common now in applications, letters, writing, because they appear in these models. The word delve is the classic example in English. I used to like the word delve. Now I don't use it in writing, partly because I think people will think I'm using a language model. But it turns out that a lot of the reinforcement learning was done in Nigeria, and the particular version of English they use in Nigeria happens to include the word delve as a very commonly recurring word. So one of the reasons these models say delve a lot of applications I now get with letters use the word delve, is because people in Nigeria trained these models to be more human-like, and the words that they use include the word delve. Anyway, none of that quite answers your question. They put a lot of other guardrails around to try and make sure that the models were much more conservative, but I think the real answer is I think the data was just much, much larger.
Yeah, so I mean, we also start using more and more AI models, and I think what we find is the challenge is that often the data quality and the data quality is not good enough for some of the things we want to do. For example, we use it in Parkinson's disease. We have progression models, we have genetics from the patients, but there's just not enough genetics of patients that have been followed carefully enough over time. And so it is getting there, but I think it's just going to be a matter of time now for actually generating the right data to feed the AI system. And so it's an iterative process, like you said, the patient talking to the AI and force it back. I think it's also with regard to data creation. We need to kind of figure out what are the most appropriate data that can help us to use AI in the best way to actually make real breakthroughs.
Yeah, I think that's a really important point, and one worth making. These emergent properties where the models start to appear human really only arise with massive data. If you and I think the equivalent in, let's say, the world of DNA and RNA or epigenetic models, let's say for AI, we're going to need really massive data to get to the point of seeing these emergent corporate property equivalents that we now see in kind of language.
Can I just ask also, how much of a barrier is access to quality data for the future of AI?
Yeah, I think it's critical, and an even bigger issue might even be the access to compute, because there is a GPU shortage, one of the reasons that NVIDIA has had this very sharp rise, and that there's a lot of concern over where the chips are made and who designs them. From a kind of geopolitical perspective, is that the models are really dependent on both data size and then chips for training. and both of those are becoming a more scarce resource. The tech companies may be the only ones. This is one of the bullets I put at the end. Maybe there's a handful of tech companies that are actually capable of having the money. First of all, some of these models cost $5 billion to train, so it's not the sort of thing my lab can do. Just as an example, or any of us probably here, even pulling our grants on stage, you're not going to get to that kind of money. So then you need access to data. An organization like Google, for example, has all of YouTube as well, so they can actually go into every video on YouTube, pull down the text from video to text or speech to text, and then use that to train. So they have a hidden advantage there, if you like, already. So data size and compute together are going to be the limiting factors.
Ivan, you have any question?
No. I just want to update for this young audience. Many U.S. universities, at least I know Yale, has been putting a lot of effort to investing in AI. We created a $500 million budget. One point of the budget is to address the data issue, especially medical AI. There are lots of confidential information, and how good AI is depends on how good data you're feeding to them. And we are building our even own the AI cloud, so to speak, a close network with 5 other Eastern Coast universities. And we use hydro plant, green energy.
Yeah, I think that's actually a really important point I was going to make it a moment ago. If we leave the power just in the hands of the tech companies, they're ultimately, their aim is to improve shareholder value. Right. Of course, that often results in good things for the world, but not always. And so I think it's really important that academic institutions come together, because that's the only way they would manage to be able to build compute resources they can all use, or academic or not-for-profit institutions, so that the models aren't entirely controlled in the private sector.
So now come one realistic problem for the students. If they like to study bioscience, should they also learn computer as well? I mean, look like that you cannot be an excellent scientist without certain knowledge of an AI expert, just like yourself.
Yeah, I think it is actually a really good idea to have at least some basic knowledge of coding. It's partly a reflection of what my lab looks like, but it's now been many years since I had a pure wet lab scientist come to the lab. I often have mostly wet lab scientists, but even I spoke to one new student this week, and I felt that maybe she had done less computing than most of the folks in the lab, but even she would say, well, actually, in my spare time, I've been learning Python, and I'm looking, I can do Bash, and I can do some of my own analysis, and I want to learn to be able to do more. So I do think it's almost like a fundamental skill now in a wet lab to be able to deal with higher throughput data and be able to do at least some basic scripting. The good news is, I didn't talk about this, that almost nobody anymore writes code without a co-pilot next to them, which is to say a language model writing the code or correcting the code. For the last 2 years, literally nobody in my lab has written code without a co-pilot or language model helping them do it.
So I think for all of you in the audience, I'm sure you must have all been very impressed about the passion by which our speakers have talked about their research. So truly, to become a scientist, a life scientist, obviously you have to have a passion. So I'd like to ask each one of you, who obviously have a passion, to answer very briefly, what is the major problem you would like to solve going forwards now? You've talked a lot about what you've achieved, but can you just briefly say what would be the major problem that you would like to solve going forward? So maybe start with Anne.
Putting me on the spot.
Ladies first.
Well, I'm a geneticist, and I think one of the conceptual advances that we really need to understand is about variation within the population, and in particular to classic genetic concepts around, they're called variable penetrance and variable expressivity. This is how I alluded to this here, trying to understand why a bunch of people with the same genetic mutation have different manifestations of that mutation, and indeed how offspring that inherit that mutation have different manifestations from each other. So what is the explanation of variation within of particular diseases. And a lot of that will have to do, I think, with regulatory molecules that are acting at these loci, so-called trans factors, many of which will probably be epigenetic.
Thank you, Anne. So maybe Haifan, you'd like to have another go?
Okay, sure. Since I was the last speaker, you probably still remember a few of my words. My last question really is to understand how the entire genome function is integrated together. Just remember that our genes are just few percent of the genome. The vast majority of the genome is essentially still unknown. And without knowing such important parts of the genome, I don't think we are understanding the biology so completely. And in the case of these pRNAs, even though we just started discovering it, many of these pRNAs and the PV proteins themselves now have been shown to be essential in many types of cancers. That indicates there are even missing cancer mechanisms from the basic understanding level that we don't know. And I hope this will be my question, how the genome is integrated and how pRNA mediate this integration. And with tens of millions of pRNAs, I need AI's help. And then together, how can that benefit the clinical application? That's my next question.
Thank you, Haifan. And Lorenz?
Yeah, I still remember when I got this same question when I got hired at Sloan Kettering from Jim Rossman, who went on to win the Nobel Prize for Wiesner, Tiesner's. And I remember I was sitting in the taxi and told him, oh, there's two things I really would like to do. One is really understanding how you make all the cell types from pluripotent cells, and the other thing is, to cure Parkinson's disease. So we didn't really completely get there, but we're getting closer now to actually having an impact. But I think the next two things for me is actually this issue that I alluded to, this timing question. So what really controls time? So why do we get maximum 90 or 100 years old? What controls lifespan? What controls early timing? What are the factors? And I think there's still a huge mystery. We understand very well transcription networks at an early stage, but time is still kind of a mystery. And the other thing is very ambitious, I guess, is really focusing more and more actually on Alzheimer's disease, which is extremely complex because it's not just dopamine neurons, there's many neurons. And we actually think currently that maybe there's some really innovative ways for how we can use microglial cells there. And so that's maybe the goal for maybe the next 10 or 20 years.
Yeah. Your next frontier. So Euan, what's your next frontier?
Yeah, I think I would answer in relation to the genome part of my lab and maybe the last slide I had put up there, because I think that we've gotten so much better at sequencing the genome to be able to diagnose disease. We'd love to understand variable penetrance best. So you got that one for us. You got that one solved. We want to be able to also treat and do genome engineering to cure those diseases. And there are some genetic diseases that, for now, that's a reasonable word to use, effective cures. But to do that, we've got to be able to get those really amazing new tools, base editors, prime editors, CRISPR tools, to the right cell at the right time. And so I would love to solve delivery. I remember talking to Andy Fire, his name came up. So I called him up when I was quite new at Stanford, and I had this idea about allele-specific silencing for these diseases, and we did some work in that arena. I asked him, what were the major issues that we need to solve, and he said, there's 5 things you need to solve: delivery, 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 and delivery. So we haven't solved it yet, so we'd love to try and solve it.
Taking science from a slightly different aspect, in Hong Kong, recently we had great promotion of entrepreneurship, trying to transform science into a commercial business. I do see that a lot of successful cases here. So I'd like to hear, what is your journey like, from a good scientist into a good entrepreneur and make it into a so-called strong business? So start with Lorenz. I know that your company was bought by Bayer just a few years ago. So sure. How was the journey like?
I think it was a journey that kind of naturally or emerged from our work. So it was really the passion of actually making a difference. And you can only do that up to a certain level on an academic scale. We talked about billions of dollars now of AI network models. You're just not going to get a grant for that. No? Yeah. And so you need to kind of have a different way to raise funds. And that was really for us the limiting factor. We had a big grant, $15 million was big for us from the state of New York to get all the way to make the cells ready for the clinic. But then it finished. And so it is, first of all, we need to have more money, but also if we really want to treat millions of people, there are more than 10 million of people with Parkinson's disease, that's never going to happen without really scaling it up. And so we wanted to have a big partner. And it was the time when stem cells really were kind of at the high time. No, not many people have tried it at that entrepreneurial level. So we really dreamed at the time now to make what we call the Genentech of cell therapy. So we had like a cardiac program, a neuro program, and actually some other programs as well. And I think it's going to trim down on some few companies that succeed, but I think it's just this next level of the dream. So I think many people ask me, so why do you then not go into the company? But I think that's for me not as attractive because so many people do that. It's a huge effort, and there are other people that do that probably better. But to give it the push, to give the idea the push to actually turn into a company, that's for me one of the most exciting parts. And then go back to the lab now and work on the next big idea.
You, maybe I take the angle slightly different. You and I sit on the same company, the AstraZeneca board of director. How do big companies like this take the science into so-called the patient, but it's not just for profit, but also try to make a difference to the humankind?
Yeah, I mean, I think at the end of the day, we've talked a little bit about this almost like academic versus industry perspective, but the reality is that the people who go into work every day in those companies, whether it's a small biotech or a big pharma company, aren't going in saying, I wonder how I can make more money today. I mean, there are people, I'm sure, on the finance side of those companies who do that, but the scientists aren't doing that. And for the most part, they want to change the world the same way everyone else does. They want to cure disease. And so I think that journey is, and you know, beautifully described here and with this really lovely work, it is a journey. And so many things begin, even for big pharma, like AstraZeneca, like Bayer, you know, it begins in small labs, often in academia, sometimes in small biotechs. At some point, it has to leave academia, and then sometimes postdocs go out, sometimes the PIs go out too, and continue that journey. It's certainly very fun to be able to take that journey with the idea of whether you remain in academia or not, and then eventually, potentially, into a very big pharma company that can really distribute it to the world. It's I think, very worthwhile.
Thank you. Anne?
Yeah, I would echo that. The key thing, I think, is about porosity. It's about the movement of people from different sectors in a smooth and comfortable way. And I think actually AstraZeneca epitomizes that really beautifully in the way that they've integrated into my ecosystem at the University of Cambridge, where AstraZeneca just landed slap bang in the middle of our academic environment. And I think at the beginning, everybody was sort of, oh my goodness, you know, there was a little bit of concern about what this would look like. But actually, it was quite remarkable watching within a very short period of time the trust that was built up between academics and our industrial colleagues who were really excited about talking to academics, and that brought researchers into that industry landscape in a very, very quite remarkable way. And then the third thing, I think, so there's this porosity, there's the synthesis of an enabling ecosystem. You know, we in Cambridge, I mean, certainly on the east and the west coast of the US, they're doing this absolutely beautifully and seamlessly. But that end-to-end journey from fundamental research into its application and commercialization is really having an ecosystem where that is visible to young people coming into the system is really, really important. And then the final thing, which I am very excited about because I see our young people doing this much better than my generation, is that not being afraid to take risks, that, you know, wanting to try something and it being okay if it doesn't work. And then moving on and trying something else. It's aligned with the porosity, moving from academia into industry, back into academia again, setting up a small company, it may be failing, setting up another one that might fail not quite so badly, and then starting another one that works, based on interacting with colleagues, being supported by academics, engaging with industrial colleagues that are in the landscape. So it's porosity, creating an enabling landscape, and not being afraid to take risks.
So taking that note, I think that our time is almost up. To the student, I would like to say that life science is great fun. It's an excellent journey that you can travel through from all the way from, you know, the knowledge of the biology to AI to commercialization, and then it's actually a very rewarding experience. So, Kathy, would you like to make a final word?
one final word from you, Haifan.
Okay.
Okay. What do you think then? Is it necessary for all basic scientists to make that journey, to make the journey all the way to what often governments want, commercialization and companies?
Well, my simple answer is not all necessary. Everyone should pursue your passion. And whatever you're passionate about, if it's basic research, that's enough for you, stay there. If your passion is to start a biotech company, then go with that passion, follow that passion. For example, at Yale, we have students, including actually one student in my lab. She started the Yale Biotech Club. Basically, it's an apprenticeship kind of organization that allows students to get into biotech. And there's a whole big place called the Yale Entrepreneurial Club that link with business school. But I see only about maybe 25% of students truly had that passion. And for those students who have that passion, they did really well. But if you're just there, just to try out your life or something, be prepared that, there are lots of other opportunities you can consider. And my only advice is follow your passion in research.
Well, on those very, very wise words, so we say thank you to our panel, and thank you to all the audience, and I hope the young people in the audience, you enjoyed that session, and hopefully you'll be inspired to take up life sciences.
Thank you very much. Thanks a lot for the insightful sharing. May I invite all the guests on stage to have a photo together. Could you please rise, and then we can have a photo together? Okay, so let's get ready, and then don't forget your big smile. Have a look to the center, please. One more to go. 1, 2, 3. 1, 2, 3. 1, 2, 3. Okay, one more. One, two, three. 1, 2, 3. Okay, thank you so much. Thank you for the sharing. It's really insightful. And please be seated. So now it's the finale of today's symposium. Ladies and gentlemen, once again, thank you so much for joining our 2024 Future Science Prize Week and the Science Symposium. Tomorrow's topic will cover physics, mathematics, and computer science. I hope you enjoyed today's program as much as I did. For those who have registered for the Hong Kong Science Museum Guide Tour, please follow the instructions from our helpers. For those who will be joining the welcome dinner, please remember to gather in front of the entrance at 4:55, as we have arranged a shuttle bus for you. See you around during the Future Science Prize Week. Thank you.